Have you ever wondered what is happening inside you at this very moment as you lie perfectly still? While your room is quiet and your breath has slowed to a rhythmic hum, there is a vibrant, invisible symphony playing within every corner of your being. Trillions of tiny workers are building, repairing, and communicating, all using the elegant language of molecules. This is the story of how you are made. And tonight, we are going to learn that language together. In this first lesson, we are stepping into the foundational world of biochemistry. It is the bridge between the physical laws of the universe and the spark of life itself. We are moving beyond just seeing the body as a collection of organs, and instead viewing it as a masterpiece of chemical engineering. If you are ready to begin this journey of discovery, perhaps consider subscribing to our channel so we can continue this quiet exploration together every night. Now, let us settle in and begin our study. Lesson 1. Introduction to the Chemistry of Life. In our previous discussions about the world around us, we often looked at things we could touch and see. But tonight we take our first deliberate step into the microscopic. In this lesson, we are laying the groundwork for everything that follows. We are going to explore the beautiful intersection where biology meets chemistry. I want you to think of this as the introduction to the chemistry of life. It is here that we find the answers to how inanimate atoms, things like carbon, oxygen, and hydrogen, come together in a specific, logical dance to create a living, breathing, thinking person. We aren't just looking at chemicals in a jar. We are looking at the very essence of your existence. Together, we will see how the laws of the universe apply to the cells in your body, and we will begin to identify the primary characters in this story, the great macromolecules that build our world. To truly understand biochemistry, we must first appreciate what the word itself means. It is the study of the chemical processes within and relating to living organisms. It is a unique field because it tells us that life is not a magical departure from the laws of physics, but rather a sophisticated application of them. Imagine, for a moment, a grand city. To understand how that city functions, you could look at the maps or the traffic, but eventually you would need to look at the bricks, the mortar, and the electricity flowing through the wires. In the city of your body, biochemistry is the study of those bricks and that electricity. At the heart of this entire field is a single, remarkable element: carbon. In the world of chemistry, I often think of carbon as a wise and social diplomat. It sits in a special place on the periodic table that allows it to form 4 strong bonds with other atoms. Because it can reach out in 4 different directions at once, it can build structures of incredible complexity, long chains, intricate rings, and branching trees of molecules. Without carbon's unique ability to hold hands with so many others, the complex molecules required for life simply couldn't exist. Take a moment to let that idea find its place. Your entire physical form is built upon the social nature of a single type of atom. As these carbon atoms link together, they form the skeletons for what we call macromolecules. Macro simply means large, and these are the giant molecules that do the heavy lifting in your cells. There are 4 main families of these molecules that we will get to know very well: carbohydrates, lipids, proteins, and nucleic acids. Think of carbohydrates not just as food, but as the primary energy currency and structural support of the living world. They are the quick-burning fuel that keeps your lights on. Then we have lipids, which you might know as fats. In the classroom of biochemistry, we see lipids as the architects of boundaries. They create the walls and membranes that keep the inside of a cell safe from the outside. Next, we have proteins, which are perhaps the most versatile of all. If the cell is a construction site, the proteins are the workers, the tools, and the scaffolding all at once. They speed up reactions, they move cargo, and they provide the very shape of your muscles. And finally, there are the nucleic acids, like DNA and RNA. These are the master blueprints, the archives of information that tell the proteins how to build the rest of you. If your mind is still following along, try visualizing these four families working in perfect harmony. The carbohydrates provide the power, the lipids build the rooms, the proteins do the chores, and the nucleic acids hold the instructions. This is the fundamental logic of the chemistry of life. It is orderly, it is efficient, and it is happening right now within you, with no effort required on your part. To understand how we came to know these truths, we have to look back at a time when people thought life was governed by a mysterious vital force that was completely separate from the laws of chemistry. For centuries, it was believed that the molecules found in living things could never be recreated in a laboratory because they possessed a spark that inanimate matter lacked. This was known as vitalism. However, in 1828, a patient and curious chemist named Friedrich Wöhler changed everything. He was working with a mineral substance called ammonium cyanate, something entirely non-living. Through his experiments, he accidentally transformed this mineral into urea, a compound that was well known to be a byproduct of animal metabolism. This was a revolutionary moment in human history. Wöhler had shown that the chemistry of life and the chemistry of rocks were actually the same thing. He proved that we could understand life by studying the same atoms found in the stars and the earth. This discovery opened the door for modern biochemistry. It allowed us to see the cell as a chemical machine. When we look deeper into the structure of these molecules, we see that they are built using a beautiful principle called polymerism. Most macromolecules are polymers, which comes from the Greek words for many parts. Imagine a long freight train. Each individual car is a monomer, or a single part. When you link these cars together, you get a polymer. For example, a protein is a long train, where the individual cars are amino acids. A complex carbohydrate, like the starch in a potato, is a train where the cars are simple sugar molecules. The way these cars are linked is through a process called dehydration synthesis. It sounds complicated, but it's quite poetic. To join two molecules together, the cell removes a tiny molecule of water. As if the two molecules are shedding a tear to create a bond that holds them together. To break them apart later, the cell simply adds a molecule of water back in, a process we call hydrolysis. Don't worry about memorizing these terms. Just let the logic of it wash over you. The building and breaking of life is as simple as adding or removing water. This is a good place to rest your thoughts for a moment before we continue. Think about the incredible history behind this knowledge, how we moved from believing in magic forces to understanding the precise clockwork mechanisms of molecular bonding. We are part of a long lineage of thinkers who wanted to know what we are truly made of. You might wonder why we spend our time studying these microscopic bonds and ancient experiments. The reason is that biochemistry is the foundation of all modern medicine and nutrition. When you feel a burst of energy after a meal, you are experiencing the breakdown of carbohydrate polymers into monomers that your cells can burn. When you see a wound heal on your skin, you are witnessing your nucleic acids directing proteins to build new lipid membranes and structural fibers. So you see, this isn't just a theory in a book. It's the principle that explains why we need to eat, how we grow, and even how we dream. Every medication a doctor prescribes is designed to interact with this specific chemistry. If we know the shape of a protein, we can design a medicine that fits into it like a key into a lock. Biochemistry gives us the power to repair the machinery of life when it falters. It also changes how we see ourselves in the universe. It reminds us that we are made of the same fundamental materials as everything else. The carbon in your DNA was once inside a star that exploded billions of years ago. We are, quite literally, organized stardust. Understanding the chemistry of life is a way of recognizing our deep connection to the cosmos. It turns the dry facts of science into a story of belonging. Now that we have introduced the main characters of our story, the carbon atoms and the 4 great families of macromolecules, we have a sense of the stuff that life is made of. We have seen that life follows a logical chemical plan. But these molecules don't exist in a vacuum. They are constantly moving, floating, and reacting within a very specific environment. Having now grasped the basic structure and history of these building blocks, we are perfectly prepared to appreciate the medium that allows them to function. Life as we know it cannot happen in a desert of dry atoms. It requires a solvent, a gentle cradle to hold the chemistry together. So, when we return in our next lesson, we will begin our study of the most essential, most miraculous liquid in the universe. We will explore the properties of water and why it is the only place where the dance of biochemistry can truly begin. Let that thought rest in your mind as you drift deeper into relaxation. You are made of stars, held together by water, and driven by a chemistry that is as old as time itself. Sleep well, and we will continue our journey when you are ready. Lesson 2. Water, the solvent of life. In our previous session, we laid the critical groundwork by exploring the carbon-based architecture that defines the biological world. We saw how atoms of carbon link together to form the sturdy skeletons of life. But as we move forward in our journey, we must recognize that these molecules do not exist in a vacuum. They do not operate in a dry, static environment. For the chemistry of life to truly begin, for those molecules to move, collide, and react, they require a medium that is as dynamic as they are. This brings us to the next natural step in our curriculum, the study of water. In a university syllabus, you would find this chapter titled Water, the Solvent of Life, and for good reason. Water is the silent liquid cradle of every biochemical reaction. It is the most abundant molecule in any living cell. And tonight, we are going to explore why its seemingly simple structure is actually a masterpiece of molecular engineering. We will see how its unique shape allows it to act as a solvent, a temperature regulator, and a literal glue that holds the microscopic world together. To understand water, we must look closely at its structure, which is far more elegant than the simple formula H2O might suggest. A single molecule of water consists of one oxygen atom bonded to two hydrogen atoms. However, it is the way they are arranged that changes everything. Instead of sitting in a straight line, the oxygen atom sits at the top, with the two hydrogens angled downward, creating a shape that looks somewhat like a wide V. In a formal academic setting, we would note that the bond angle is approximately 104.5 degrees. This specific geometry is caused by the oxygen atom's unshared electrons, which push the hydrogen atoms away, forcing them into that bent position. But the shape is only half the story. The true secret of water lies in a concept called electronegativity. You can think of electronegativity as a tug of war for electrons. In our water molecule, the oxygen atom is much more greedy for electrons than the hydrogen atoms are. Because the oxygen pulls those negative electrons closer to itself, it develops a slight negative charge, which we represent with the symbol delta minus. Conversely, the hydrogen atoms, having their electrons pulled away, are left with a slight positive charge, or delta plus. This separation of charges means that water is a polar molecule. It has a positive pole and a negative pole, much like a tiny bar magnet. Take a moment to let that idea find its place. Every drop of water in the world is composed of billions of these tiny magnets, all pulling and pushing on one another in a constant rhythmic dance. Because water molecules are polar, they are incredibly sticky. The positive hydrogen end of one molecule is naturally attracted to the negative oxygen end of its neighbor. This attraction creates what we call a hydrogen bond. Now, in the grand hierarchy of chemistry, a single hydrogen bond is quite weak. It is easily broken and easily reformed. But when you have billions of them working together, they create a collective strength that is formidable. This stickiness gives rise to two critical properties: cohesion and adhesion. Cohesion is water's tendency to stick to itself, which is why raindrops form perfect spheres. Adhesion is its tendency to stick to other surfaces, which is how water is able to climb up the narrow vessels of a tall tree, defying gravity to reach the highest leaves. If your mind is still following along, try visualizing this. Imagine a vast, crowded room where everyone is briefly holding hands with the people next to them, letting go, and then reaching out for someone else. This is the fluid, ever-changing network of hydrogen bonds that defines liquid water. It is this constant hand-holding that allows water to remain a liquid at temperatures where other molecules of similar size would have long since turned into gas. As we go deeper into the why of water, we encounter its most famous title, the universal solvent. And in the history of science, early alchemists spent lifetimes searching for a menstruum universale, a substance that could dissolve anything. While water cannot dissolve everything, it comes closer than any other liquid on Earth. Because of its polarity, water is exceptionally good at dissolving ionic compounds, like common table salt, and other polar molecules, like sugar. When you drop a crystal of salt into water, the water molecules surround the individual sodium and chlorine ions. The negative oxygen ends of the water molecules huddle around the positive sodium, while the positive hydrogen ends surround the negative chlorine. They gently pull the crystal apart, encasing each ion in a hydration shell. This ability is fundamental to your survival. It is what allows your blood to carry dissolved minerals, vitamins, and wastes through your veins. Without this solvent property, the interior of your cells would be a dry, motionless collection of salts and proteins, unable to interact or sustain life. Another remarkable layer of water's character is its high specific heat. In our previous lesson, we mentioned that biochemistry requires stability. Water provides this stability by acting as a thermal buffer. Because of those hydrogen bonds we discussed, it takes a great deal of energy to make water molecules move faster and heat up. The water must first absorb enough energy to break those sticky bonds before the temperature can rise. This is why the ocean stays cool even on a blistering summer day, and it is why your body temperature doesn't spike the moment you step out into the sun. Water protects you from the volatility of the outside world. There is also a beautiful paradox in how water freezes. In almost every other substance in the universe, the solid form is denser than the liquid form. If you drop a solid piece of lead into liquid lead, it will sink. But water is different. As water cools toward its freezing point, the molecules slow down enough for the hydrogen bonds to become fixed and rigid. They push each other apart into a highly organized hexagonal lattice. This makes ice take up more space than liquid water, which means ice is less dense. This is why ice floats. Historically, this anomalous property of water is what allowed life to survive during the ice ages. If ice sank, lakes and oceans would freeze from the bottom up, eventually becoming solid blocks of ice that never thawed, crushing all life within them. Instead, the ice forms a protective, insulating blanket on the surface, allowing the liquid water and the life within it to remain safe and relatively warm underneath. When we step back and look at the big picture, we see that you are essentially a walking, talking container of water. Roughly 60 to 70% of your body mass is this singular liquid. This isn't just a random fact of biology. It is the very principle that makes your physiology possible. Think about the act of sweating on a warm afternoon. This is a direct application of water's high heat of vaporization. As you sweat, your body uses its internal heat to break the hydrogen bonds of the water on your skin. When those water molecules finally break free and evaporate into the air, they take that heat with them, leaving you cooler. It is a cooling system that relies entirely on the molecular stickiness we have been studying. So you see, water isn't just a background setting for life. It is an active participant. It determines the shape of your proteins, the structure of your cell membranes, and the speed of your metabolism. Even the way your DNA stays twisted in its double helix is partly due to how its parts hide from or seek out the surrounding water. This is why NASA's search for life on other planets usually begins with one simple question: is there evidence of liquid water? We have yet to find a single form of life that can function without it. It is the common denominator of all biological existence. We have spent this time appreciating the fluid stage upon which the drama of life is performed. We have seen how the simple polarity of a water molecule creates the bonds, the solvent power, and the thermal stability that we often take for granted. We have looked at the cradle. Now, it is time to look at what is being cradled. Let's hold that thought here. A complete piece of the puzzle. We now understand the medium, the solvent of life. Our next lesson will hand us the adjacent piece, which involves the complex machinery that floats within this water. We are going to move from the liquid environment to the primary structural workers of the cell. We will begin our study of the building blocks of muscle, enzymes, and the very fabric of our physical being. When we return, we will explore the elegant world of amino acids and the primary structure of proteins. Take a deep breath and let the stillness of the water we've discussed settle over your mind. Your body is doing this work for you right now, maintaining its inner sea in perfect balance while you rest. Lesson 3. Amino acids and the primary structure of proteins. In our last session, we spent time appreciating the gentle rhythmic nature of water, the liquid cradle that holds all of life's secrets. We saw how its polarity creates a stable environment where molecules can meet and interact. Now that we understand the stage upon which life performs, it is time to meet the leading actors. In any university biochemistry curriculum, this is where we begin our deep study of the macromolecules, starting with the most diverse and hardworking of them all, the proteins. But to understand a protein, we must first understand its alphabet. Just as a grand novel is composed of simple letters, a protein is composed of small, elegant units called amino acids. In this lesson, are titled Amino Acids and the Primary Structure of Proteins. We are going to look at the anatomy of these building blocks and learn how they link together to form the very first level of protein organization. We are moving from the environment of the cell to the actual machinery of your being. If you were to look at a protein under a powerful microscopic lens, you would see a long, winding chain. That chain is made of amino acids. Although there are hundreds of amino acids in nature, the standard set that builds almost all life on Earth consists of just 20. I find it beautiful that the immense diversity of life, from the silk of a spider's web to the hemoglobin carrying oxygen in your blood, is written with an alphabet of only 20 letters. Every amino acid shares a common foundational anatomy. At the very center sits a single carbon atom, which we call the alpha carbon. Because carbon is that social diplomat we discussed in our first lesson, it reaches out in 4 directions to hold 4 different partners. To one side, it holds a hydrogen atom. To another, it holds an amino group, which is a nitrogen atom bonded to 2 hydrogens, NH2. To the third side, it holds a carboxyl group, COOH, which gives the molecule its acidic property. Together, these parts form the backbone of the amino acid. This backbone is identical in nearly every amino acid you will ever encounter. But it is the 4th partner that makes each amino acid unique. This is the side chain, often labeled simply as the R group. If the backbone is the stationary part of a building, the R group is the personality of the room. In some amino acids, the R group is just a single hydrogen atom, making the molecule small and nimble. In others, it is a complex ring of carbons that makes the molecule bulky and rigid. Some R groups are hydrophobic, meaning they fear water and try to hide away from it, while others are hydrophilic, reaching out to embrace the water molecules surrounding them. Take a moment to let that idea find its place. Your body is built from 20 different personalities, some shy, some outgoing, some large and some small, all waiting to be linked together. When the cell decides to build a protein, it must link these amino acids together in a specific order. This happens through a beautiful chemical handshake called a peptide bond. To form this bond, the carboxyl group of one amino acid meets the amino group of the next. As they join, they release a tiny molecule of water, that tear of connection we mentioned before, in a process called dehydration synthesis. This bond is remarkably strong and surprisingly rigid. It creates a peptide backbone that can grow and grow until it forms a long string called a polypeptide. The specific linear sequence of these amino acids, the exact order in which they are linked from the first to the last, is what we call the primary structure. This sequence is not random. It is dictated exactly by your genetic code. If your DNA is the master architect's blueprint, the primary structure is the first row of bricks laid down according to those plans. It is a simple, one-dimensional string, but it contains all the information necessary for the protein to eventually become a three-dimensional machine. And to truly appreciate the elegance of the primary structure, we should look back at the history of how we discovered it. For a long time, scientists weren't sure if proteins had a fixed, specific sequence. It wasn't until the 1950s that Frederick Sanger, a two-time Nobel Prize winner, spent years painstakingly determining the exact sequence of amino acids in the hormone insulin. He proved that every molecule of insulin in every human being is made of the exact same sequence of 51 amino acids. This was a turning point in biochemistry, showing us that life is not a vague soup, but a precision-engineered system. If your mind is still following along, try visualizing the vastness of the possibilities. Even with a small protein of only 100 amino acids, the number of ways you could arrange those 20 different R groups is greater than the number of atoms in the entire observable universe. Yet, out of all those trillions of possibilities, your body chooses the exact one it needs to make your heart beat or your eyes see. There is another layer of detail we must consider: the concept of chirality. Almost all amino acids are chiral, meaning they can exist in two forms that are mirror images of each other, much like your left and right hands. In the vast majority of living organisms on Earth, only the L form, the left-handed version, is used to build proteins. Why life chose the left hand over the right billions of years ago remains one of the great, quiet mysteries of science. It is as if the universe decided on a specific handshake, and every living thing has honored that tradition ever since. We also categorize these 20 amino acids based on whether our bodies can make them from scratch. You may have heard the term essential amino acids. These are the ones your body cannot manufacture on its own. They are the letters of the alphabet that you must import through the food you eat. There are 9 of these essential building blocks, and they remind us of our deep physical connection to the rest of the biological world. We rely on other living things to provide the very letters we use to write the story of our own lives. Don't worry about memorizing the names like leucine, tryptophan, or glutamine right now. Just think of them as specialized tools. Some are used to create the tight, decoiled structure of a hair strand, while others are designed to sit in the center of an enzyme and grab onto a specific chemical target. The primary structure is the setup for everything that is to come. Every twist, every fold, and every function of the protein is hidden within that simple, linear sequence. It can be easy to view the primary structure as just a list of names, but its importance becomes very clear when we look at what happens when even one letter is out of place. Consider the condition known as sickle cell anemia. This life-altering condition is caused by a change in just one single amino acid in the primary structure of hemoglobin, a protein that contains hundreds of amino acids. Because one hydrophobic amino acid replaces a hydrophilic one, the entire protein begins to behave differently when oxygen is low. It causes the hemoglobin molecules to stick together, distorting the shape of the red blood cell. So you see, this isn't just a theory in a book. The precision of the primary structure is what keeps us healthy. It is the reason why a specific enzyme in your stomach can digest your dinner while leaving your own tissues untouched. The order of the amino acids determines the identity of the protein. This is also why nutrition is so vital. When you consume protein, whether from a glass of milk, a handful of nuts, or a piece of fish, your body doesn't just keep that protein as it is. It uses the hydrolysis we discussed earlier, adding water back in to break the peptide bonds and harvest the individual amino acids. These amino acids then enter a pool in your body, waiting for your DNA to call them back into service to build something new. You are constantly breaking down old books to write new ones. You are a living library that is being continuously updated, letter by letter. We have now seen how the alphabet of life is constructed. We've looked at the central carbon, the common backbone, and the diverse R groups that give each amino acid its unique character. We've watched as they join hands through peptide bonds to form the primary structure, the long, elegant string of potential. But a long string is not yet a machine. Just as a thread must be woven into a fabric to become a garment, these polypeptide chains must be folded into specific shapes to become functional. A protein's power comes not just from its sequence, but from its geometry. Having now grasped the structure of the individual building blocks, we are perfectly prepared to appreciate how they begin to interact with one another. Our next lesson will hand us the adjacent piece of the puzzle, where we see these long chains begin to twist into spirals and fold into pleated sheets. This is where the simple string becomes a three-dimensional masterpiece. When we return, we will explore protein folding and three-dimensional structure. This is a good place to rest your thoughts for a moment. Imagine that long, beautiful chain of amino acids within you, perfectly sequenced and ready for the next stage of its journey. You are a work of art in progress even as you sleep. I'll be here to guide you through the next fold when you are ready. Lesson 4. Protein folding and three-dimensional structure. In our last session together, we marveled at the alphabet of life, those 20 amino acids that link hands to form the primary structure of a protein. We saw them as a long linear string, a sequence of characters carrying a specific message from your genetic code. But in the world of biochemistry, a string is just a beginning. To become a functioning tool, a machine, or a structural support, that string must undergo a dramatic transformation. Imagine a long, thin ribbon of silk. On its own, it can do very little, but when folded with precision and care, it can become a beautiful piece of origami, or a strong, supportive fabric. This is the essence of our fourth lesson: protein folding and three-dimensional structure. We are moving from the one-dimensional sequence to the three-dimensional reality. We will explore how these chains twist, turn, and collapse into the shapes that define your very existence. It is here, in the geometry of the molecule, where the magic of biology truly takes form. When we speak of protein folding, we are describing the process by which a polypeptide chain finds its unique functional shape. This isn't a chaotic or random jumbling of the chain. Rather, it is a highly organized hierarchy of levels. We have already discussed the primary structure, the sequence itself. Now, we must look at the secondary structure. This level of folding doesn't actually involve the unique R groups or side chains we talked about before. Instead, and it relies on the repetitive nature of the polypeptide backbone itself. The atoms in the backbone, the nitrogens and the oxygens, begin to form hydrogen bonds with one another across the gaps of the chain. This creates two very famous shapes. The first is the alpha helix. Picture a delicate spiral staircase or a coiled telephone cord. This shape is held together by hydrogen bonds that form every 4 amino acids, creating a sturdy, flexible rod. The second shape is the beta pleated sheet. Imagine taking a piece of paper and folding it back and forth to create a series of ridges and valleys, like a paper fan. These sheets are incredibly strong and provide the structural foundation for many proteins. Take a moment to let that idea find its place. Your body is full of these microscopic spirals and fans, thousands of them, clicking into place with mathematical precision. Once these secondary structures are established, the protein takes a massive leap into the tertiary structure. This is the final 3-dimensional fold of a single polypeptide chain. While the secondary structure was about the backbone, the tertiary structure is all about the R groups, those 20 different personalities we met in Lesson 3. These side chains start to interact with one another across much larger distances. Imagine a group of friends at a crowded party. Some friends are naturally attracted to one another. They form ionic bonds, or hydrogen bonds, to stay close. Others, like the sulfur-containing amino acids, form disulfide bridges, which are like strong permanent staples that lock parts of the protein together. But perhaps the most powerful force of all is the hydrophobic effect. Many of the R groups are oily and water-fearing. Because they are surrounded by the water we studied in Lesson 2, they naturally scrunched together in the center of the protein to hide from the moisture. This inward collapse is what ultimately tucks the protein into its final, compact shape. If your mind is still following along, try visualizing this. The protein is like a long piece of wire. The secondary structure gives the wire some curls and zigzags, but the tertiary structure is when the wire is finally bent into the shape of a specific key, ready to unlock a specific door in your body. To truly appreciate the mystery of folding, we should consider a fascinating concept known as Levinthal's paradox. In the 1960s, a scientist named Cyrus Levinthal pointed out something startling. If a protein tried to find its correct fold by simply trying every possible shape at random, it would take longer than the age of the entire universe to fold just one single protein. Yet, in your body, proteins fold in mere milliseconds. This tells us that the folding process is not a random search, but a directed pathway. The protein wants to reach its lowest energy state, the most comfortable, relaxed position possible. In the history of this science, we must also look at the work of Christian Finsen. In a series of famous experiments, he showed that if you unfold a protein using chemicals, a process we call denaturation, and then remove those chemicals, the protein will often fold itself back up into its original, correct shape all on its own. This led to Finsen's dogma, the idea that all the information needed for a protein to find its 3D shape is contained within its primary sequence. The sequence is the destiny. However, Sometimes the journey to the correct shape is difficult. The inside of a cell is a crowded, busy place, and a folding protein can easily get tangled or stuck to its neighbors. To prevent this, the cell uses specialized proteins called chaperones. I like to think of these as the patient mentors of the molecular world. Some chaperones, called chaperonins, are shaped like tiny hollow barrels. A misfolded or struggling protein can enter this barrel, where it is shielded from the chaos of the cell, giving it the peace and quiet it needs to fold correctly before being released back into the environment. Don't worry about the complex physics of thermodynamics or entropy right now. Just think of the protein as an explorer looking for the most peaceful valley to rest in. It is always seeking balance, always looking for the shape that allows it to be at peace with its surroundings. This drive toward stability is one of the most fundamental laws of nature, and it is happening inside you at an unimaginable scale. The importance of this biological origami cannot be overstated because in biochemistry, shape is function. If a protein does not fold into its precise three-dimensional shape, it cannot do its job. It would be like trying to turn a bolt with a wrench that hasn't been cast into the right size. The tool is simply useless. When proteins fail to fold correctly, or when they unfold due to heat or changes in pH, the consequences are significant. You see this every time you cook an egg. The clear liquid protein in the egg white is full of neatly folded soluble proteins. When you add heat, you provide enough energy to break the weak bonds holding those folds together. The proteins denature, tangling into a chaotic white, solid mess. This is denaturation in action, a permanent change in shape that renders the original tools of the egg white unusable. In our own bodies, misfolded proteins are more than just a culinary curiosity. They are at the heart of many health challenges. Conditions like Alzheimer's disease, Parkinson's, and prion diseases, like mad cow disease, are caused by proteins that have folded into the wrong shape. These misfolded proteins often become sticky, clumping together into plaques that interfere with the normal function of brain cells. In the case of prions, a misfolded protein can actually act like a bad influence, touching a correctly folded protein and causing it to misfold as well. It is a reminder of how delicate the balance of life truly is, and how much we depend on the precision of these molecular folds. So you see, this isn't just an abstract lesson in geometry. Every breath you take, every thought you form, and every beat of your heart depends on the fact that your proteins are holding their shapes. Your body is a masterpiece of architectural stability, maintained by the constant quiet work of folding and the watchful eyes of the chaperone proteins. We have now traveled from the simple string of amino acids to the complex, three-dimensional masterpiece of the folded protein. We've seen how the backbone creates spirals and sheets, and how the R groups interact to tuck the protein into its final functional form. We have learned that shape is the key to everything a protein can do. But now that we have our tools built and shaped, we must ask, what do these tools actually do? How do they perform the work of life at such incredible speeds? Having now grasped the architecture of the protein, we are perfectly prepared to see these shapes in action. Our next lesson will hand us the adjacent piece of the puzzle, where we look at a very specific and vital class of proteins. These are the movers and shakers of the cell. The proteins that don't just sit there but actively drive chemical reactions forward. We are going to explore the world of enzyme kinetics and catalysis. We will see how a protein's shape creates a special pocket, where the chemistry of life is accelerated to lightning speeds. This is a good place to rest your thoughts for a moment. Picture those thousands of proteins within you, each holding its shape with quiet dignity, ready for the work of the coming day. You are a structure built of structural integrity, right down to your very molecules. I'll be here to guide you through the first of those chemical reactions when you are ready. Lesson 5. Enzyme Kinetics and Catalysis. In our previous session, we watched as the long linear strings of amino acids transformed into intricate, three-dimensional masterpieces. We learned that for a protein, shape is everything. It is the destiny of its function. But now that we have these beautifully folded tools, we must ask ourselves what they actually do within the bustling city of the cell. If the structural proteins we discussed are the scaffolding and the bricks, then the proteins we are about to meet are the master craftsmen, the engines, and the accelerators of life. In a university biochemistry curriculum, this chapter is titled Enzyme Kinetics and Catalysis, and it represents one of the most awe-inspiring parts of our study. We are going to explore how life manages to perform complex chemical reactions at lightning speed, reactions that, in a simple beaker of water, might take hundreds of years to occur. We will see how an enzyme's shape creates a sacred space for chemistry to happen and how we can measure the incredible speed of life. It is a journey from the static form of the protein to the dynamic movement of metabolism. To understand an enzyme, we must first understand the concept of a catalyst. In the world of chemistry, a catalyst is any substance that increases the rate of a reaction without being consumed by the reaction itself. Imagine you are trying to move a heavy boulder over a steep hill. On your own, you might struggle for hours, pushing with all your might, perhaps never reaching the top. An enzyme acts as a team of workers who come along and dig a tunnel straight through the base of that hill. The destination is the same, but the effort required to get there is drastically reduced. In biochemistry, that hill is what we call the activation energy, often denoted as E sub A. Every chemical reaction requires a little push to get started, a spark to break old bonds so new ones can form. Enzymes work by lowering this activation energy, making it much easier for the reaction to slide forward. The secret to this power lies in a very specific part of the enzyme's folded shape, the active site. If you remember our discussion on protein folding, you'll recall how the R groups of different amino acids are tucked into specific positions. In an enzyme, several of these R groups come together to form a specialized pocket or groove. This is the active site, and it is perfectly designed to welcome a specific molecule called the substrate. I like to think of the active site as a master craftsman's workbench, perfectly contoured to hold the exact piece of wood he is working on. For a long time, scientists used the lock and key model to describe this. They thought the substrate fit into the enzyme as perfectly and rigidly as a key fits into a deadbolt. However, we now prefer a more graceful explanation called the induced fit model. Imagine a glove waiting for a hand. The glove has a general shape, but as your hand slides inside, the glove stretches and molds itself to the unique contours of your fingers. When the substrate enters the active site, the enzyme undergoes a subtle shift in shape, hugging the substrate. This gentle squeeze puts physical stress on the substrate's chemical bonds, stretching them until they are ready to snap and rearrange. Take a moment to let that idea find its place. Your body is currently performing trillions of these molecular hugs, each one precisely timed to keep your heart beating and your mind clear. Once the reaction is complete, the substrate has been transformed into a product. Because the enzyme hasn't been changed by this process, it simply lets go of the product and returns to its original shape, ready to greet the next substrate molecule. This is a cycle that can repeat thousands or even millions of times every single second. It is a continuous rhythmic process of greeting, transforming, and releasing. When we move beyond the simple how of enzymes, we enter the world of kinetics, the study of how fast these reactions happen. This is the realm of two brilliant scientists from the early 20th century, Leonore Michaelis and Maud Menten. Maud Menten, in particular, was a pioneer, one of the first Canadian women to earn a medical degree. She traveled to Berlin to collaborate on the equations that still bear her name today. Together, they developed the Michaelis-Menten equation, which helps us describe the speed limit of an enzyme. We measure this speed using two primary values. The first is Vmax, which represents the maximum velocity of the reaction. Imagine a small post office with only one clerk. If only one person is waiting in line, the clerk can work at a relaxed pace. As more people arrive, the clerk works faster and faster, but eventually, the clerk reaches their absolute limit. No matter how many hundreds of people join the line, the clerk can only process them so fast. This is Vmax, the point where every single enzyme molecule is saturated with substrate and working at full capacity. The second value is Km, the Michaelis constant. This value tells us about the affinity, or the attraction, between the enzyme and its substrate. A low Km means the enzyme is like a powerful magnet. It can find and grab its substrate, even if there are only a few molecules floating around. A high Km means the enzyme is a bit more indifferent. It needs a high concentration of substrate before it really starts working effectively. Don't worry about the math involved. Just picture it as a measure of how eager an enzyme is to get to work. If your mind is still following along, you try visualizing the complexity of regulating these speeds. The cell doesn't always want its enzymes running at Vmax. Sometimes, it needs to slow down a process to save energy or prevent the buildup of too much product. To do this, the cell uses inhibitors. There are competitive inhibitors, which are molecules that look almost exactly like the substrate. They sneak into the active site and sit there, blocking the door so the real substrate can't get in. Then, there are the more subtle, non-competitive, or allosteric inhibitors. These don't go for the active site at all. Instead, they attach to a completely different part of the enzyme, the backdoor, if you will. When they attach, they cause the entire enzyme to shift its shape slightly, just enough so that the active site on the other side no longer fits the substrate. It is as if someone leaned against the back of the glove we discussed earlier, twisting it so your hand no longer fits inside. This is a beautiful, elegant way for the cell to turn its machinery on and off as needed. This is a good place to rest your thoughts for a moment before we continue. Think about the incredible precision of these movements. We often think of chemistry as random collisions, But enzymes turn that randomness into a directed, governed symphony. Every reaction has its place, its speed, and its off switch. You might be surprised to realize how often you interact with enzyme kinetics in your daily life. If you have ever used a biological laundry detergent to get a grass stain out of a shirt, you are using enzymes called proteases that have been designed to eat the proteins in the stain. These enzymes are engineered to have a high Vmax and to work effectively at the specific temperatures of a washing machine. In the world of medicine, enzymes are the primary targets for many of the most important drugs we have ever discovered. Aspirin, for instance, works by acting as an inhibitor for an enzyme called cyclooxygenase, which produces the molecules that signal pain and inflammation in your body. By blocking the workbench of that enzyme, aspirin prevents the pain signals from being built in the first place. Similarly, many life-saving medications for blood pressure or cholesterol are simply sophisticated keys designed to sit in the active sites of specific enzymes, slowing down a process that has become too fast or out of balance. So you see, this isn't just a theory in a book. It's the principle that allows us to treat diseases and understand how our own bodies recover. It explains why a fever can be dangerous, because heat provides so much energy that it can start to vibrate the enzyme's delicate folds until they denature, losing that precious active site. Everything we are, from the way we taste our food to the way we fight off a virus, is a result of these tireless molecular catalysts. Enzymes also remind us of the incredible efficiency of nature. A single molecule of the enzyme carbonic anhydrase in your blood can process 600,000 molecules of carbon dioxide every single second. Humans have yet to build any machine that can match that level of speed, precision, and longevity. We are built of the most advanced technology in the known universe, and it is a technology made of folded handshakes and hugs. We have now explored the machinery of life in its most active form. We've seen how enzymes lower the barriers to reaction, how they welcome substrates into their active sites, and how we can measure their incredible speed through the lens of kinetics. We've learned that the cell is not just a collection of parts, but a governed system of regulated speeds and shapes. But even the most efficient machine needs a source of power. A craftsman cannot work without energy, and an enzyme cannot build without the raw materials of life. Having now grasped the function of the protein-based machinery, we are perfectly prepared to appreciate the fuel that keeps those machines running. Our next lesson will hand us the adjacent piece of the puzzle, where we turn our attention away from the workers and toward the energy. We are going to explore the sweet, essential world of carbohydrates. We will look at how simple sugars like glucose provide the immediate spark for our enzymes to do their work, and how complex starches act as the long-term storage for the coming days. When we return, we will begin our study of carbohydrates, structure, and function. Let that thought rest in your mind. While you are sleeping, your enzymes are still at work, repairing your tissues and processing the energy of the day, moving with a rhythm that is as steady as your heartbeat. I'll be here to show you the fuel for that rhythm when you are ready. Lesson 6. Carbohydrates, Structure and Function. In our previous session, we watched the incredible machinery of the cell, the enzymes, as they performed their rapid rhythmic work. We saw how their unique shapes allowed them to catalyze the reactions that keep us alive. But even the most finely tuned machine requires a steady supply of fuel to operate, and a craftsman needs raw materials to build new structures. In this sixth step of our journey, we turn our attention to the molecules that provide that spark and that substance. In a university curriculum, this is the study of carbohydrates' structure and function. You might know them simply as carbs or sugars. But in the quiet world of biochemistry, they are so much more than just a source of energy. They are the carbon-based skeletons of our world, the identification tags on our cells, and the primary way life stores the sun's power. We are going to explore the elegant hierarchy of these molecules, from the simple sweetness of a single sugar ring to the massive, complex chains that make up the sturdy trunk of an oak tree, or the energy reserves in your own liver. To begin our study of carbohydrates, let us look at the name itself. Carbohydrate literally means watered carbon. If you were to look at the chemical formula of a basic carbohydrate, you would see a very predictable ratio. For every one carbon atom, there are 2 hydrogen atoms and 1 oxygen atom. We often write this as CH2O sub N. It is as if life took a simple carbon atom and added the components of a water molecule to it. This simple building block is the foundation for everything that follows. We categorize carbohydrates based on how many of these building blocks are linked together. The simplest unit is the monosaccharide. The word saccharide comes from the Greek word for sugar, and mono means one. These are the simple sugars, and the most famous of these is glucose. Glucose is a 6-carbon sugar, shaped like a hexagonal ring, and it is the universal currency of biological energy. It is the primary molecule your brain craves as you sleep. There are others, of course, like fructose, which is found in fruit, and galactose, found in milk. Each has the same number of atoms, but they are arranged in slightly different ways, much like using the same set of bricks to build a slightly different small house. When 2 of these monosaccharides decide to join hands, they form a disaccharide. This happens through that familiar handshake we have seen before, dehydration synthesis. The 2 sugar rings link together, and a molecule of water is released. This connection is called a glycosidic bond. Common table sugar, or sucrose, is a disaccharide made of 1 glucose and 1 fructose joined together. Lactose, the sugar in milk, is a bond between glucose and galactose. These molecules are still relatively small, designed to be broken down quickly so the energy can be harvested. Take a moment to let that idea find its place. Your body is constantly assembling and disassembling these rings, snapping them together for storage, or pulling them apart for a quick burst of power. If we keep adding more and more sugar rings to the chain, we eventually create a polysaccharide. These are the complex carbohydrates. Think of a polysaccharide as a long freight train where each car is a glucose molecule. Depending on how those cars are linked, whether the train is a straight line or has many branching side tracks, the function of the molecule changes entirely. Some polysaccharides, like starch in plants or glycogen in humans, are used for energy storage. Others, like cellulose, provide structural strength. If your mind is still following along, try visualizing the difference. Starch is like a loosely coiled rope, easy for enzymes to unravel and eat when the plant needs energy. Glycogen, which you store in your liver and muscles, is even more branched. It looks like a dense, bushy shrub of glucose. Because it has so many ends, Your enzymes can snip off many glucose molecules at once, now giving you a rapid supply of energy the moment you need to wake up or run. It is a system designed for both volume and speed. To truly appreciate the depth of carbohydrate chemistry, we must look at their stereochemistry, the way these molecules sit in three-dimensional space. Even a simple sugar like glucose can exist in two different forms, a linear chain and a ring. In the watery environment of your cells, glucose spends more than 99% of its time as a ring. When that ring closes, the position of a single hydroxyl group, in -OH, can point either up or down. We call these the alpha and beta forms. This might seem like a tiny academic detail, but it is one of the most important choices in all of biology. When you link alpha glucose molecules together, you get starch or glycogen, substances we can easily digest and use for energy. But when you link beta glucose molecules together in a specific way, you get cellulose. Because of that tiny shift in the direction of the bond, cellulose forms long, straight, rigid fibers that are incredibly difficult to break. This is what makes wood strong and gives plants their skeleton. Historically, this has shaped the world around us. Human beings do not possess the enzymes necessary to break that specific beta linkage in cellulose. This is why we can eat a potato starch and get energy, but we cannot eat a tree branch and get the same result. Only certain bacteria and specialized animals like cows or termites have the key to unlock the energy hidden in cellulose. It is a beautiful example of how a microscopic change in geometry dictates the entire ecology of our planet. If your mind is wandering, try picturing those straight, strong fibers of cellulose acting as the rebar in a concrete wall. Without that specific beta bond, plants could never grow tall, and our world would look very different indeed. We should also mention the discovery of these structures. In the late 19th century, Emil Fischer, a German chemist, spent years meticulously mapping out the various shapes of sugars. He used a technique called Fischer projections to represent these 3D molecules on a 2D page. He was so successful that he won the Nobel Prize in 1902. He showed us that life is not just made of chemicals, but of specific, mirrored arrangements. Most naturally occurring sugars in our bodies are D-sugars, a term that refers to their right-handed orientation in space. Just as we saw with the left-handed amino acids, life has a preferred hand for its sugars. Don't worry about the complex math of carbon counting right now. Just remember that the cell numbers its carbons from 1 to 6. When we talk about a 1 to 4 linkage, we are simply saying that the first carbon of one sugar is holding hands with the fourth carbon of the next. It is a very orderly, very predictable system of construction. The relevance of carbohydrates is woven into every aspect of your daily life. When you hear about complex carbs being better for you than simple sugars, you are hearing a lesson in biochemistry. Simple sugars like sucrose are broken down instantly, causing a rapid spike in your blood glucose. Complex polysaccharides, like the starch in whole grains, take much longer for your enzymes to unravel, providing a slow and steady trickle of energy that keeps you feeling satisfied for hours. But carbohydrates do more than just provide fuel. They are also vital for communication. On the surface of every one of your cells, there are little trees of carbohydrates attached to proteins or lipids. We call these glycoproteins and glycolipids. These sugar chains act as a biological ID badge. They tell your immune system, I belong here, do not attack me. So you see, this isn't just a theory in a book. The most famous example of this is your blood type. Whether you are type A, B, or O is determined entirely by which specific sugar molecule is sitting on the surface of your red blood cells. A single sugar can be the difference between a life-saving blood transfusion and a dangerous reaction. Your body reads these sugars as a language of identity and belonging. This synthesis of energy and identity shows that carbohydrates are the true multitaskers of the molecular world. They store the energy of the sun in their bonds. They build the physical structures of the forest, and they provide the labels that allow your trillions of cells to work together as a single, unified person. As you rest tonight, your body is carefully managing these sugar levels, making sure your brain has exactly enough glucose to process your memories while keeping your energy reserves ready for the morning. We have now explored the sweet structural world of carbohydrates. We've seen how simple monosaccharides, like glucose, serve as the fundamental units of energy, and how polysaccharides like glycogen and cellulose build the stores and the scaffolds of life. And we have learned that a tiny change in a bond's direction can change a meal into a piece of wood. But while carbohydrates are excellent for quick energy and structural support, they have a limitation. They are very heavy because they hold on to a lot of water. For long-term storage and for creating the ultimate boundaries of the cell, the body needs something more efficient and water-resistant. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we turn our attention to the molecules that hate water but love to store energy and build walls. We are going to explore the oily, essential world of lipids. We will see how these molecules create the skin of your cells and keep your energy safe for the long term. So, when we return, we will begin our study of lipids and biological membranes. Take a deep breath and feel the steady rhythm of your own metabolism. Your sugar stores are balanced, your cells are labeled, and your body is at peace. I'll be here to show you the architects of your cell walls when you are ready. Lesson 7. Lipids and biological membranes. In our journey so far, we have explored the alphabet of proteins and the sweet, energetic architecture of carbohydrates. We have seen how life builds its machinery and its fuel. But as we move into this 7th lesson, we must address a fundamental question of existence. How does a cell know where it ends and the rest of the world begins? If a cell were merely a collection of proteins and sugars floating in the water we studied in Lesson 2, it would quickly dissolve and drift away, lost to the vastness of its environment. To exist as a distinct entity, life requires boundaries. It requires a skin, a container, and a gatekeeper. In a university biochemistry course, this chapter is titled Lipids and Biological Membranes. And it is here that we meet the architects of the cellular boundary. We are going to explore the oily, water-fearing world of lipids. We will see how these molecules, which naturally shun the water, are the only ones capable of creating the stable, flexible walls that allow life to flourish in a liquid world. To understand lipids, we must first understand their most defining characteristic. They are hydrophobic. This word, which we have touched upon briefly before, literally means water-fearing. While proteins and carbohydrates are often quite comfortable in water, lipids are non-polar. They do not have the positive and negative charges that would allow them to hold hands with water molecules. Think of a bottle of oil and vinegar salad dressing. No matter how hard you shake it, the oil eventually pulls away, huddling together in golden droplets. This natural huddling is the driving force behind the structure of every cell you possess. There are several families of lipids, but we will focus on the two that are most vital to your biology. The first is the triacylglycerol, which you might know simply as fat. A triacylglycerol consists of a 3-carbon backbone called glycerol, with 3 long fatty acid chains hanging from it like the tails of a kite. These are the molecules your body uses for long-term energy storage. They are incredibly efficient because, unlike carbohydrates, they don't carry the extra weight of water. They are the compact batteries that keep you powered during a long fast. However, the true star of this lesson is the phospholipid. This molecule is a bit of a chemical contradiction, and that is what makes it so useful. A phospholipid looks a bit like a triacylglycerol, but one of its fatty acid tails has been replaced by a phosphate group, often denoted in your textbook as PO43-. This phosphate head is polar. It loves water. Meanwhile, the 2 remaining fatty acid tails are nonpolar. They hate water. In biochemistry, we call a molecule with this split personality an amphipathic molecule. Take a moment to let that idea find its place. Imagine a molecule that is half magnet and half oil, constantly trying to satisfy 2 opposite desires at once. When you place a large number of these phospholipids into a watery environment, they solve their dilemma in a beautiful and spontaneous way. The water-fearing tails all turn inward, huddling together to hide from the moisture, while the water-loving heads turn outward to face the water. This creates a double layer of molecules that we call the phospholipid bilayer. This bilayer is the fundamental structure of all biological membranes. It is a thin, oily film that creates a continuous barrier. It is strong enough to hold the cell together, yet flexible enough to allow the cell to move, grow, and divide. If your mind is still following along, try visualizing this bilayer not as a rigid wall, but as a sea of oil. It is only 2 molecules thick, so thin that it would take 10,000 of them stacked on top of each other to equal the thickness of a single piece of paper. Yet, this delicate film is the skin that protects the delicate chemistry of your life from the chaos of the outside world. To truly appreciate the elegance of the cell membrane, we must look at a concept introduced in 1972 by scientists S.J. Singer and Garth Nicholson, the fluid mosaic model. They proposed that the membrane is not a static, solid shell, but a dynamic, shifting mosaic. Imagine a vast swimming pool filled with hundreds of thousands of ping-pong balls. These balls represent the phospholipids. They are constantly jiggling, sliding past one another, and even spinning in place. Because they are not chemically bonded to their neighbors, only held together by their mutual fear of water, they are free to move. This fluidity is essential for life. Floating in this oily sea are proteins, which we can think of as the icebergs. Some of these proteins sit on the surface, while others, called integral proteins, span the entire width of the membrane. These proteins are the gatekeepers. They are the channels that allow specific nutrients to enter and wastes to leave. And they are the receptors that catch chemical messages from other parts of the body. Without these proteins, the membrane would be a closed wall. With them, it becomes a sophisticated communication hub. There is another layer of detail we must consider: the kinks in the fatty acid tails. In your study of nutrition, you have likely heard of saturated and unsaturated fats. In biochemistry, this refers to the presence of double bonds between carbon atoms. A saturated fatty acid has no double bonds. Its tail is straight and can pack tightly against its neighbors, much like bricks in a wall. This makes the membrane more rigid. An unsaturated fatty acid, however, has a cis double bond that creates a sharp bend, or a kink, in the tail. If your mind is still following along, try visualizing this. May these kinks act like little elbows that push the neighboring molecules away, preventing them from packing too tightly. This is a good place to rest your thoughts for a moment. This kinking is how your cells regulate their fluidity. If the environment gets too cold, the membrane might risk freezing and becoming brittle. To prevent this, the cell adds more unsaturated lipids with more kinks to keep the oil flowing. Your body also uses a molecule called cholesterol as a fluidity buffer. Cholesterol sits between the phospholipids, preventing them from huddling too close in the cold, but also holding them together if things get too warm. It is a masterpiece of self-regulation. Historically, the study of lipids was often overshadowed by the glamour of DNA and proteins. But as we have learned more about how cells communicate, we have realized that lipids are much more than just fat. They are active signaling molecules. Some lipids can be clipped off the membrane and sent into the center of the cell to act as a second messenger, telling the nucleus to turn a gene on or off. The membrane is not just a container, it is a sensory organ for the cell. The importance of lipids and membranes is visible in every aspect of your health and daily life. Think about the vitamins you take. You may have noticed that vitamins A, D, E, and K are labeled as fat-soluble. This means they are hydrophobic, just like the lipids in your membranes. Because they share the same chemical nature, these vitamins can dissolve right into your cell walls, where they can be stored for long periods. This is why you don't need to consume them every single day, unlike the water-soluble vitamins like C or B, which pass through your system much more quickly. So you see, this isn't just a theory in a book. The nature of lipids explains why some medicines are applied as a cream on the skin. The oily base of the cream helps the medicine melt through the lipid bilayers of your skin cells and enter your body. It also explains why soap is so effective at cleaning. A soap molecule is also amphipathic. It has a tail that grabs onto the oils and dirt on your hands, and a head that grabs onto the water. When you rinse, the soap pulls the oil away into the water, leaving your skin clean. On a deeper level, the integrity of your membranes is what allows your nerves to fire. Your brain is the most lipid-rich organ in your body. The long extensions of your nerve cells are wrapped in a thick, fatty coating called myelin. This myelin acts exactly like the plastic insulation on an electrical wire, preventing the electrical signal from leaking out and allowing it to travel at incredible speeds. When you think a thought or wiggle a toe, you are relying on the insulating power of lipids. As you rest tonight, your body is carefully maintaining these trillions of oily boundaries. It is checking the fluidity of your membranes, repairing any leaks, and ensuring that each cell remains a sovereign kingdom, protected by its own shimmering fluid wall. You are a collection of boundaries, held together by the quiet, persistent power of the phospholipid. We have now explored the oily, protective world of lipids and the dynamic, shifting mosaic of the cell membrane. We've seen how the simple fear of water can create the complex structures that define the edges of life. We have seen the skin of the cell and the icebergs of protein that float within it. But a cell with a wall and an engine still needs a master plan. A city with walls and workers still needs a library where the blueprints are kept and a way to pass those plans on to the next generation. Having now grasped the physical boundaries and the material structures of life, we are perfectly prepared to appreciate the most famous molecule of all. Let's hold that thought here. A complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we turn our attention away from the stuff of the cell and toward the information. We are going to enter the nucleus and explore the incredible world of DNA and RNA, and we will see how life stores its memories and how it writes the instructions for everything we have studied so far. So when we return, we will begin our study of nucleic acids, DNA and RNA structure. Take a deep breath and let the feeling of your own physical presence, of your skin, your nerves, and your boundaries lull you into a deeper rest. Your trillions of boundaries are secure. I'll be here to show you the blueprints of your soul when you are ready. Lesson 8. Nucleic acids, DNA and RNA structure. We have spent our previous sessions wandering through the physical landscapes of the cell, admiring the liquid sea of water that cradles life, the intricate protein machinery that performs its labor, and the shimmering lipid boundaries that define its edges. But as we stand here at the 8th step of our journey, we must ask, where does the wisdom come from? A city can have walls, and it can have workers, but without a central archive, a library where the blueprints are kept, and the history of the city is recorded, it would quickly fall into chaos. In a university biochemistry curriculum, this is the moment we enter the sanctum of the nucleus to study nucleic acids, DNA and RNA structure. These are the molecules of information. They are the master blueprints that tell the proteins how to fold and the cell how to grow. Tonight, we are going to look at the elegant, twisting architecture of these molecules. We will see how a simple sequence of chemicals can store the entire complexity of a human life, and how the subtle differences between DNA and RNA allow life to both preserve its past and communicate its future. To understand nucleic acids, we must start with their smallest unit, the monomer known as the nucleotide. Just as an amino acid is the building block of a protein, the nucleotide is the building block of the genetic code. Every nucleotide is a 3-part masterpiece of chemical engineering. At its heart sits a 5-carbon sugar, which we call a pentose. Attached to one side of this sugar is a phosphate group, and to the other side is a nitrogenous base. And I often like to think of the sugar and the phosphate as the hardware of the molecule. They form the sturdy repeating railing of the staircase, while the nitrogenous base is the software, the actual letter of the code. There are 2 main families of these nitrogenous bases. First, we have the purines, which are adenine and guanine. These are the larger double-ringed structures. Then, we have the pyrimidines, which are cytosine, thymine, and uracil. These are smaller, single-ringed structures. In the world of DNA, the 4 letters we use are A, T, C, and G. In RNA, the thymine is replaced by uracil. This might seem like a small substitution, but it is one of the ways the cell distinguishes between its permanent archives and its temporary messages. Take a moment to let that idea find its place. You are built from an alphabet of only 4 letters, repeated and arranged in a sequence billions of characters long. When these nucleotides link together, they form a sugar-phosphate backbone. The phosphate of one nucleotide holds hands with the sugar of the next through a very strong connection called a phosphodiester bond. This creates a long, continuous strand. In deoxyribonucleic acid, or DNA, 2 of these strands come together to form the famous double helix. Imagine a flexible ladder that has been gently twisted into a spiral. The railings of the ladder are the sugar-phosphate backbones, and the rungs of the ladder are the nitrogenous bases reaching out to meet each other in the center. The way these bases meet is governed by a strict and beautiful law: complementary base pairing. Because of the way their hydrogen bonds are arranged, adenine always pairs with thymine, and cytosine always pairs with guanine. A purine always meets a pyrimidine. This ensures that the rungs of our DNA ladder are always exactly the same width, keeping the helix perfectly symmetrical. It also means that each strand contains the information of the other. If you know the sequence of one side, you can perfectly predict the other. This is the secret to how life copies itself so accurately. If your mind is still following along, try visualizing the difference with ribonucleic acid, or RNA. While DNA is a double-stranded, stable, and permanent spiral, RNA is typically single-stranded and much more flexible. RNA also uses a slightly different sugar called ribose, which has one more oxygen atom than the deoxyribose found in DNA. That single oxygen atom makes RNA more reactive and less stable, which is perfect for its role as a temporary messenger that can be easily created and destroyed by the cell as needed. To truly appreciate the structure of DNA, we must look at how it is oriented in space. In a university setting, we speak of the directionality of the strands. Because of the way the carbon atoms in the sugar ring are numbered, one end of a DNA strand is called the 5' end, and the other is the 3' end. In a double helix, the 2 strands are anti-parallel. This means they run in opposite directions, like a 2-lane highway. One strand runs 5 feet to 3 feet, while its partner runs 3 feet to 5 feet. This orientation is critical because the enzymes that read and copy your DNA can only move in one direction. The history of this discovery is one of the most famous stories in all of science. In the early 1950s, many scientists were racing to find the structure of the hereditary molecule. While James Watson and Francis Crick are often the names we remember, their work was built upon the essential foundations laid by others. There were Chargaff's rules, which noted that in any organism, the amount of adenine always equals the amount of thymine, and the amount of cytosine always equals guanine. And most importantly, there was the work of Rosalind Franklin. Franklin was an expert in X-ray crystallography. She captured a famous image known as Photo 51, which provided the mathematical proof that DNA was a helix of a specific width. When you look at a DNA model today, and with its major grooves and minor grooves, the wider and narrower gaps in the twisting spiral, you are seeing the physical reality that Franklin's X-rays first revealed. These grooves are not just for show. They are the windows through which proteins can read the bases hidden inside without having to unzip the whole molecule. If your mind is wandering, try to picture the scale of this. If you were to take the DNA from just one of your cells and stretch it out in a straight line, it would be about 2 meters long. Yet your body manages to fold and coil this massive thread so tightly that it fits inside a nucleus only a few millionths of a meter wide. It does this by wrapping the DNA around spools made of proteins called histones. This combination of DNA and protein is called chromatin. It is an incredible feat of microscopic packaging, ensuring that the blueprints are always accessible but never tangled. Don't worry about the complex geometry of the 5' and 3' ends right now. Just think of it as the reading direction of a book. Life is a story that is always read from left to right, or in this case, from the 5' end to the 3' end. It is a system of profound order, maintained across billions of years of evolution. The structure of nucleic acids is the foundation for everything we understand about inheritance disease and the diversity of life. Every physical trait you possess, the color of your eyes, the shape of your smile, even your tendency to stay up late or wake up early, is encoded in the sequence of those 4 simple bases. When we talk about the human genome, we are talking about the complete set of these instructions, a library that contains about 3 billion rungs on the DNA ladder. So you see, this isn't just a theory in a book. It is the principle that allows for forensic science, where a single strand of hair can be used to identify a person with perfect certainty because their DNA sequence is unique to them. It is the basis for modern medicine, such as mRNA vaccines, which work by sending a temporary message into your cells, written in the language of RNA, to teach your immune system how to recognize a specific virus. The universality of this code is perhaps its most moving aspect. The same 4 bases that make up your DNA also make up the DNA of a redwood tree, a soaring eagle, and the tiny bacteria living in the soil. We are all part of the same chemical conversation. This shared language is what allowed scientists to take a gene from a jellyfish that makes it glow and place it into a bacterium and have it work perfectly. The bacterium read the jellyfish's instructions because the alphabet is the same. As you rest tonight, your DNA is quietly holding the secret of who you are. It is the silent, stable anchor of your identity. While your proteins are being recycled and your lipids are shifting in their fluid membranes, your DNA remains, a constant and faithful record of your life's design. You are a biological masterpiece, written in a language that is both ancient and ever new. We have now explored the profound twisting library of the nucleic acids. We've seen how the nucleotides form the letters, how the sugar phosphate backbone forms the railings, and in how the antiparallel strands create the stable double helix of DNA. We have seen the difference between the permanent records of DNA and the nimble single-stranded messages of RNA. But a library, no matter how vast its knowledge, is a cold and quiet place without energy. Blueprints cannot build themselves, and messages cannot be sent without power. To move from the information of the nucleus to the action of the cell, we must understand how life harvests the energy it needs to read the code and build the machines. Having now grasped the information and the structure of life, we are perfectly prepared to appreciate the fire that drives it all. Our next lesson will hand us the adjacent piece of the puzzle. where we turn our attention to the great engine of the cell. We are going to enter the world of metabolism and bioenergetics. We will see how life takes the energy from the universe and transforms it into the currency of ATP to power everything we have studied so far. So when we return, we will begin our study of Introduction to Metabolism and Bioenergetics. Take a deep breath and let the knowledge of your own internal library bring you a sense of peace. You are perfectly designed, and your story is being kept safe within every cell of your body. I'll be here to show you how that story is powered when you are ready. Lesson 9. Introduction to Metabolism and Bioenergetics. In our journey together thus far, we have looked at the exquisite architecture of life. We have studied the water that provides the stage, the proteins that serve as the machinery, the lipids that define the boundaries, and the nucleic acids that hold the master blueprints. We have the stuff and the plans, but a city with beautiful buildings and detailed blueprints is still a ghost town if the lights are off and the machines are silent. To bring this microscopic world to life, we need a flow of energy. In a university biochemistry curriculum, this is the pivotal moment where we enter the world of metabolism and bioenergetics. We are going to explore the grand internal economy of the cell, how it earns, spends, and saves its energy. We will move beyond the static shapes of molecules and begin to see the cell as a dynamic, roaring fire of activity. It is a story of how life defies the natural tendency of the universe to fall into chaos, using a tiny molecule called ATP as its universal currency. To understand metabolism, we must first look at it as the sum total of every single chemical reaction happening within an organism. It is not just one process. It is a vast interconnected web of thousands of different pathways, all running simultaneously. We generally divide this immense complexity into two broad categories: catabolism and anabolism. I find it helpful to think of these as the breaking down and the building up of life. Catabolism is the set of metabolic pathways that break down larger, complex molecules, like the carbohydrates and fats we have studied, into smaller units. As these large structures are dismantled, energy is released. Imagine a grand old stone bridge being carefully deconstructed. As each stone is removed, the structural energy that held it up is let go. Anabolism is the exact opposite. It is the set of pathways that use that released energy to construct the complex components of the cell, such as new proteins or strands of DNA. It is the building of a new house using the materials and energy gathered from elsewhere. Together, catabolism and anabolism create a continuous cycle, a rhythmic ebb and flow that defines the metabolic state. Take a moment to let that idea find its place. Your body is a constant construction site, where the old is being recycled to power the new, in a balance so perfect that you rarely ever feel it happening. But how exactly is this energy moved from the breaking down side to the building up side? In the world of human economics, we use money so that a farmer can trade wheat for a pair of shoes without having to carry a sack of grain to the cobbler. In the cellular economy, that money is a molecule called adenosine triphosphate, or ATP. Biological diagram shows structure of adenosine triphosphate, ATP. ATP is a remarkable piece of molecular engineering. It consists of an adenosine base, a ribose sugar, and a chain of 3 phosphate groups. The The magic happens in that chain of phosphates. Because these phosphate groups are all negatively charged, they naturally want to repel one another, much like three powerful magnets forced together. We can imagine this chain as a tightly coiled metal spring held back by a delicate latch. When the cell needs energy to move a muscle or build a protein, it pulls the latch by breaking off the last phosphate group through a process called hydrolysis. This releases a burst of energy and leaves behind ADP, or adenosine diphosphate. To recharge the battery, the cell uses the energy from the food you eat to snap that third phosphate back on, turning ADP back into ATP. If your mind is still following along, try visualizing the scale of this. Your body does not store a massive vault of ATP. Instead, it recycles it at a staggering rate. A single molecule of ATP is typically spent and recharged thousands of times every single day. If you could see your own internal economy, you would see a flurry of activity where batteries are being drained to do work and immediately sent back to the mitochondrial power plants to be filled again. It is a system of profound efficiency, ensuring that energy is always exactly where it needs to be, right when it is needed. To truly appreciate the why behind metabolism, we must look at the laws that govern the entire universe, the laws of thermodynamics. Bioenergetics is essentially the application of these laws to living systems. The first law tells us that energy cannot be created or destroyed, only transformed. When you eat a piece of fruit, you are not creating energy. You are simply transforming the chemical energy stored in the sugar bonds, energy that originally came from the sun, into the mechanical energy of your movements, or the thermal energy that keeps you warm. The second law of thermodynamics is perhaps more profound for the study of life. It states that the universe is constantly moving toward a state of increased disorder or entropy. Left to its own devices, everything falls apart. A tidy room becomes dusty. A garden becomes overgrown with weeds. A mountain eventually crumbles into sand. Life, however, is a defiant stand against this law. By using a constant stream of energy, your cells maintain an incredible level of order and complexity. You are like a person constantly sweeping a floor in a windstorm. The moment you stop spending energy, entropy takes over. In the history of biochemistry, we owe much of our understanding of this energy balance to Josiah Willard Gibbs, an American scientist who developed the concept of free energy, often denoted as G. We look at the change in free energy, or delta G, to determine if a reaction will happen spontaneously. If a reaction releases energy, it has a negative delta G and is called an exergonic reaction. These are the downhill reactions of catabolism. If a reaction requires an input of energy, it has a positive delta G and is called an endergonic reaction. These are the uphill reactions of anabolism. The genius of the cell lies in energy coupling. The cell takes a downhill reaction that releases a lot of energy, usually the hydrolysis of ATP, and physically links it to an uphill reaction that needs energy. I like to think of this like a water wheel. The water naturally wants to fall downhill, the exergonic reaction, by placing a wheel in the path of that falling water, and we can use its movement to grind grain or generate electricity, the endergonic work. In your cells, enzymes are the wheels that facilitate this coupling, ensuring that the energy from ATP isn't just wasted as heat, but is used to perform the vital tasks of life. Don't worry about the complex math of Gibbs free energy right now. Just think of it as a way of measuring the willingness of a reaction to occur. Life is a collection of thousands of tiny energy transactions, each one calculated to keep the flame of your existence burning bright against the cold pull of entropy. The principles of bioenergetics are not just abstract concepts for a textbook. They are the very things you feel every day. When you feel tired or low on energy, you are experiencing a moment where your rate of ATP production is struggling to keep up with your metabolic demands. When you shiver in the cold, your body is intentionally triggering inefficient metabolic reactions that release a lot of energy as heat rather than storing it, to keep your core temperature stable. Even as you lie here now, drifting towards sleep, your metabolism remains highly active. We call this your basal metabolic rate, or BMR. It is the minimum amount of energy your body needs just to keep the lights on, to keep your heart beating, your lungs expanding, and your brain cells maintaining their delicate electrical balance. In fact, your brain is one of the most expensive organs you own, consuming about 20% of your daily ATP, despite being only a small fraction of your body weight. So you see, this isn't just a theory in a book. This energy economy is what connects you to every other living thing on this planet. Whether it is a deep-sea vent bacteria or a soaring hawk, every organism uses the same fundamental strategy. They harvest energy from their environment, they convert it into the currency of ATP, and they spend that currency to fight back against chaos. We are all part of a grand cosmic exchange of energy. This synthesis of chemistry and physics reminds us of the preciousness of life. Every thought you have and every breath you take is a hard-won victory against the second law of thermodynamics. You are an island of order in a sea of disorder, maintained by the constant, quiet work of your metabolic pathways. It is a beautiful, invisible effort that your body performs for you every second of your life. We have now explored the grand overview of metabolism and the energetic money of the cell. We've seen how catabolism and anabolism balance each other, how ATP acts as a rechargeable battery, and how life uses the laws of thermodynamics to maintain its own complex order. We have seen the economy of the cell and the fire that drives it. But knowing that we need energy is only the first half of the story. The second half is knowing exactly how we get it. We know that ATP is the currency, but how do we actually earn it from the food we eat? How does a molecule of sugar from your dinner become a burst of power for your cells? Having now grasped the broad principles of bioenergetics, we are perfectly prepared to look at the very first step in this energy harvesting process. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we turn our attention to an ancient and universal pathway. We are going to look at how the cell takes a single molecule of glucose and begins to break it down in a 10-step process that happens in the cytoplasm of every cell you own. We are going to explore the world of glycolysis. So when we return, we will begin our study of glycolysis and the catabolism of glucose. Take a deep, slow breath and feel the warmth of your own internal fire. Your metabolism is keeping you safe and steady while you rest, managing your energy with the wisdom of billions of years of evolution. I'll be here to show you how you turn your fuel into fire when you are ready. Lesson 10. Glycolysis and the catabolism of glucose. In our last session, we spent some time discussing the grand economy of the cell. we looked at the concept of metabolism as a whole, and we introduced the remarkable molecule ATP as the universal currency that allows life to pay for its many complicated tasks. But knowing that ATP exists is only half the story. To truly understand the wonder of your own biology, we must look at how that currency is actually earned. We must move from the bank to the factory floor. In a standard university biochemistry curriculum, this is where we begin our study of glycolysis and the catabolism of glucose. This is an ancient, beautiful pathway, perhaps one of the oldest chemical sequences on our planet. It is the process by which a single molecule of sugar is carefully, step by step, dismantled to harvest the energy hidden within its bonds. It is a journey that happens not in some specialized organelle, but in the very cytoplasm of every single cell you own. Whether you are a tiny bacterium in the soil or a human being resting in a warm bed, the logic of glycolysis remains the same. Tonight we are going to walk through the 10 purposeful steps of this pathway. We will see how the cell invests energy to make energy, and how it transforms a simple sugar into the building blocks for everything that follows. To understand glycolysis, let us start with the name itself. Glyco refers to glucose, our 6-carbon sugar, and lysis means to split or to break apart. At its simplest level, glycolysis is the story of how a cell takes one 6-carbon molecule of glucose and splits it into 2 3-carbon molecules called pyruvate. This process is entirely anaerobic, meaning it does not require oxygen to function. This is a testament to its ancient origins, dating back to a time on Earth before our atmosphere was rich with the oxygen we breathe today. Imagine, if you will, a long assembly line inside the fluid of your cell. At the beginning of the line, a single molecule of glucose enters. Over the course of 10 distinct steps, each managed by its own specific enzyme, this molecule is twisted, rearranged, and eventually cut in half. We can think of these 10 steps as being divided into 2 major acts. The first act is the energy investment phase, and the second act is the energy payoff phase. In the energy investment phase, the cell actually spends 2 molecules of ATP to get the process started. It might seem strange for a cell to use up its precious currency when the goal is to make energy, but I want you to think of this as a start-up cost. Much like you might need to spend money to buy tools for a new job, the cell must prime the glucose molecule to make it reactive enough to break. By attaching phosphate groups to the ends of the sugar, the cell makes the molecule unstable. It energizes it, preparing it for the big split. Take a moment to let that idea find its place. Even at the molecular level, there is a rhythm of investment and patience before the reward. Once the glucose has been primed and split into 2 3-carbon pieces, we enter the energy payoff phase. This is where the cell reaps the benefits of its earlier investment. In these final steps, the cell harvests high-energy electrons, which are tucked away into a carrier molecule called NADH. More importantly, the cell produces 4 molecules of ATP. Because we spent 2 at the beginning and made 4 at the end, we are left with a net gain of 2 ATP molecules for every 1 molecule of glucose that enters the pathway. By the end of the line, our original glucose is gone. In its place, we have 2 molecules of pyruvate, 2 molecules of NADH, and our small but vital profit of 2 ATP. The sugar has been catabolized, broken down, and its energy has been successfully captured in forms the cell can use immediately or save for later. It is a process of elegant, methodical destruction. If we look closer at these 10 steps, we find that the cell is not just randomly breaking bonds, and it is performing a highly regulated dance. One of the most important characters in this story is an enzyme called phosphofructokinase, or PFK. In the world of biochemistry, PFK is known as the pacemaker, or the gatekeeper, of glycolysis. It appears in the third step of the pathway, right before the molecule is split in half. PFK is an allosteric enzyme, which is a concept we touched upon in our lesson on enzymes. It has a sensor that allows it to feel how much ATP is already in the cell. If the cell is resting and has plenty of ATP, the ATP molecules actually bind to PFK and slow it down. It is as if the cell says, "We have enough money in the bank. Let's slow down the factory line and save our sugar for later." Conversely, if ATP levels drop, PFK speeds up, cranking the handle of glycolysis to produce more energy. It is a perfect, self-regulating thermostat that ensures the cell never wastes its resources. There is a profound historical context to this pathway as well. Because glycolysis happens in the cytoplasm and doesn't require complex organelles like mitochondria, scientists believe it evolved nearly 3.5 billion years ago. It is the common metabolic thread that binds all life together. When we study these 10 steps, we are looking at a chemical fossil that has been preserved and perfected over eons. It is a masterpiece of efficiency that has stood the test of deep time. If your mind is still following along, try visualizing the journey of the electrons. As the sugar bonds are broken, electrons are released. But electrons cannot simply float around the cell. They are like hot coals that must be carried in a specialized bucket. That bucket is NAD+. When it picks up electrons and a proton, it becomes NADH. This NADH is a form of potential energy, a promise of much greater ATP production to come later, once we move into the mitochondria. For now, however, it is enough to know that the cell is carefully gathering every scrap of value from the sugar molecule. Don't worry about memorizing the names of all 10 enzymes, from hexokinase to pyruvate kinase. Instead, just try to feel the logic of the flow. The cell adds a phosphate, rearranges the shape, adds another phosphate, splits the molecule, and then begins the harvest. It is a systematic deconstruction, much like a master craftsman carefully taking apart an old clock to clean and reuse its gears. Every step is deliberate. Every move has a purpose. The relevance of glycolysis is something you feel in your body every single day. It's particularly when you are active, when you engage in a sudden, intense burst of movement, like sprinting for a bus or lifting something heavy. Your muscles need energy faster than your lungs can supply oxygen. In those moments, your cells rely almost entirely on glycolysis for their ATP. Because glycolysis is fast and doesn't need oxygen, it can provide a quick emergency supply of power. However, there is a trade-off. Without oxygen, the pyruvate produced at the end of the line cannot move into the mitochondria to be broken down further. Instead, it is converted into lactic acid, or lactate. This is the burn you feel in your muscles after heavy exertion. Your body is essentially taking out a metabolic loan, getting energy now at the cost of having to deal with the acidic byproduct later, once you catch your breath. So you see, this isn't just a theory in a book. It is the principle that explains why you pant after exercise. Your body is working to pay back that oxygen debt, so it can clear the lactic acid and return to a state of balance. Even more fascinating is how some of your cells rely on glycolysis exclusively. Your red blood cells, for instance, have no mitochondria. They are the only cells in your body that produce all of their energy through this ancient 10-step pathway. This is a beautiful bit of biological irony. The very cells that carry oxygen to every other part of your body do not use any of that oxygen for themselves. They are purely anaerobic, fueled by the steady, quiet hum of glycolysis as they circulate through your veins. As you lie here now, your brain and your red blood cells are consuming a steady stream of glucose. Your liver is releasing just enough sugar into your blood to keep the factory lines of glycolysis running at a perfect resting pace. You are being powered by a chemical reaction that began in the primordial seas and continues tonight within you with unfailing precision. We have now followed the journey of a glucose molecule through the 10 steps of its catabolism. We've seen the initial investment of ATP, the split into 3 carbon pieces, and the final harvest of pyruvate and energy. We have met the gatekeeper PFK and seen how our muscles use this pathway as an emergency power source. But the 2 ATP molecules we earned in glycolysis are just a small fraction of the energy hidden within the original sugar. The pyruvate we created is not the end of the story. It is merely the end of the first chapter. There is still a vast amount of energy locked inside those pyruvate molecules, waiting to be released. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we follow the pyruvate as it leaves the cytoplasm and enters the inner sanctum of the cell, the mitochondria. We are going to see what happens when oxygen is available and the cell can finally move from the emergency power of glycolysis to the high-efficiency furnace of the citric acid cycle. Having now grasped the foundation of sugar breakdown, we are perfectly prepared to appreciate the turning of the great metabolic wheel. So when we return, we will begin our study of the citric acid cycle, also known as the Krebs cycle. Take a slow, deep breath. Feel the quiet efficiency of your own cells as they process the energy of your day. You are a steady flame, burning with a logic that is both ancient and perfect. I'll be here to guide you into the heart of the mitochondria when you are ready. Lesson 11. The citric acid cycle, the Krebs cycle. We have just completed our study of glycolysis, that ancient and universal 10-step pathway that takes place in the open space of the cytoplasm. We watched as a single molecule of glucose was split into 2 molecules of pyruvate, earning us a small but vital profit of energy. But as we move into this 11th lesson, we are stepping into the inner sanctum of the cell. We are leaving the general space of the cytoplasm and crossing the double membrane of the mitochondria, the legendary powerhouse of the cell. In a university biochemistry textbook, this chapter is titled The Citric Acid Cycle, though many of us know it as the Krebs cycle, named after the man who first mapped its circular logic. It is the central engine of metabolism, a beautiful, spinning wheel of chemistry that extracts the remaining energy from the food you've eaten. Tonight, we are going to explore how this cycle turns, how it releases the carbon that you breathe out into the night air, and how it fills the electron buckets that will soon power the rest of your sleep. This is where the true harvest of energy begins. Before the cycle can even begin its first turn, we must address a small but vital transition. The pyruvate we created in the cytoplasm is a 3-carbon molecule. But the citric acid cycle is a bit exclusive. It only accepts guests that have exactly 2 carbons. Therefore, as the pyruvate enters the mitochondria, it undergoes what we call the link reaction, or pyruvate decarboxylation. An enzyme complex takes that pyruvate, snips off 1 carbon atom to be released as carbon dioxide, and then attaches the remaining 2 carbons to a shuttle molecule called coenzyme A. This creates acetyl-CoA. Think of acetyl-CoA as the golden ticket that grants entry into the cycle. It is the universal intersection where the breakdown of sugars, fats, and even some proteins all meet. Now, imagine a grand revolving door. Sitting inside that door, waiting for a guest, is a 4-carbon molecule called oxaloacetate. When the 2-carbon acetyl-CoA arrives, they join hands to form a 6-carbon molecule called citrate, or citric acid. This is the moment the cycle officially begins, and it is why we give the pathway its name. Once citrate is formed, the door begins to spin. Through a series of 8 enzymatic steps, the molecule is rearranged, pruned, and transformed, one quiet reaction at a time. As the wheel turns, the molecule is first rearranged into isocitrate. Then, in the next 2 steps, something very important happens. 2 carbons are clipped off and released as carbon dioxide, CO2. This is the very carbon that was once part of the glucose from your dinner, now ready to be carried by your blood to your lungs and exhaled. But the real prize of the cycle isn't the carbon dioxide, it's the energy. As those bonds are broken, high-energy electrons are released. These electrons are caught by our familiar carriers. NAD plus becomes NADH. After the carbons are released, the 4-carbon molecule that remains, succinyl CoA, undergoes further changes. We manage to produce 1 molecule of ATP, or its close cousin GTP, directly through a process called substrate-level phosphorylation. Then, we encounter a different electron carrier. A molecule called FAD steps in to catch more electrons, becoming FADH2. Finally, after a few more rearrangements, involving molecules called fumarate and malate, the molecule has been turned back into oxaloacetate. Take a moment to let that idea find its place. The process is a perfect loop. The oxaloacetate is right back where it started, sitting in the revolving door, ready and waiting for the next acetyl-CoA to arrive. It is a self-sustaining wheel of life that never needs to be rebuilt from scratch. It only needs to be fed. To understand the profound significance of this cycle, we must look at the man whose name it bears, Sir Hans Krebs. In the 1930s, Krebs was a young scientist who noticed that certain organic acids seemed to stimulate the respiration of cells in a way that suggested they weren't just being burned up as fuel, but were being recycled. In a moment of pure genius, he realized that the chemistry wasn't a straight line, but a circle. He published his findings in 1937, and for this, he was eventually knighted and awarded the Nobel Prize. He showed us that life doesn't just use its components. It cycles them with an elegance that far exceeds any machine built by human hands. The citric acid cycle is often described in university lectures as being amphibolic, a term that comes from the Greek word for both. This means it is both catabolic breaking things down, and anabolic, building things up. While it is famous for breaking down acetyl-CoA to make energy, it is also the central hub, where the cell can borrow molecules to build other things. If your body needs to make new amino acids for a protein or new heme for the hemoglobin in your blood, it can pull those building blocks right out of the spinning citric acid cycle. It is the ultimate multitasking center of the cell, balancing the needs of destruction and creation in every single turn. If your mind is still following along, try visualizing the electron buckets we are filling. We've mentioned NADH and FADH2 several times now, and I want you to see them as the true goal of this lesson. In glycolysis, we made a small amount of NADH, but the citric acid cycle is where we fill the majority of them. For every single molecule of glucose that started this journey, the citric acid cycle turns twice, once for each pyruvate. This produces 6 molecules of NADH and 2 of FADH2. At this stage of the journey, these molecules represent a vast amount of potential energy. I like to think of them as a bank account full of checks that haven't been cashed yet. The energy is there, locked in the movement of those electrons, waiting for the final stage of respiration to turn it into the actual cache of ATP. You might picture these carriers as little ships, heavily laden with gold, sailing through the mitochondrial matrix toward the inner membrane where the final payout awaits. The beauty of the citric acid cycle is that it connects the most basic acts of your life, the act of eating and the act of breathing. Every time you exhale into your pillow tonight, you are literally breathing out the microscopic remains of your food. The CO2 that leaves your lungs was, only hours ago, the carbon atoms inside a carbohydrate, a protein, or a fat. You are a living engine, and your breath is the gentle exhaust of 1,000 spinning chemical wheels. This cycle also explains why certain vitamins are so absolutely essential to your well-being. Many of the enzymes that drive the Krebs cycle are cofactor dependent, meaning they require B vitamins to function. For example, the shuttle, coenzyme A, is made from vitamin B5. NAD+ is derived from niacin, which is vitamin B3, and FAD comes from riboflavin, or vitamin B2. This is why a deficiency in these vitamins leads to a profound bone-deep lack of energy. Without them, the central wheel of your metabolism simply cannot turn, no matter how much fuel you provide it. So you see, this isn't just an abstract diagram in a textbook. It is the principle that explains how your body harvests the very essence of the sun's energy from your food. It is the reason why a deep, steady breath feels so revitalizing. You are providing the system with the space it needs to release its exhaust so the wheel can keep turning. It is a process of constant renewal, happening within you even as your conscious mind begins to drift away into dreams. We have now watched the great wheel of the citric acid cycle as it accepts its 2 carbon guests, spins them through 8 stages of transformation, and releases the carbon we breathe. We've seen how it harvests a little bit of immediate energy and a whole lot of potential energy in the form of those electron-filled buckets, NADH and FADH2. But as we sit here, at the end of the 11th lesson, we must acknowledge that we still haven't reached the big payout. We have all these checks written in the form of electron carriers, but the cell needs the hard currency of ATP to do its real work of repair and growth. We are standing at a precipice. The potential energy is high, the dam is full, and we are finally ready for the most spectacular stage of energy production. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we follow those electron carriers to the inner membrane of the mitochondria. We are going to witness the cellular equivalent of a massive hydroelectric dam, where a flow of protons creates a spinning molecular turbine that generates ATP at a rate we haven't seen yet. Having now grasped the turning of the wheel, we are perfectly prepared to see how that momentum is converted into power. So when we return, we will begin our study of oxidative phosphorylation and the electron transport chain. Take a long, slow, deep breath, a breath that began in the very cycle we just studied together. Your mitochondria are spinning, your electrons are being gathered, and your body is at peace. I'll be here to show you the final payout when you are ready. Lesson 12. Oxidative phosphorylation and the electron transport chain. We have traveled a long road together, following a single molecule of glucose from its first split in the cytoplasm to the spinning carbon-releasing wheel of the citric acid cycle. Along the way, we have collected several small pieces of currency in the form of ATP, but mostly we have been filling our bags with high-energy electrons. We have gathered a wealth of NADH and FADH2, which we've described as checks that have yet to be cashed. In a university biochemistry curriculum, we have finally arrived at the big payout. This lesson is titled Oxidative Phosphorylation and the Electron Transport Chain. This is the grand finale of cellular respiration, the moment where all that potential energy we've carefully harvested is converted into a flood of ATP. We are moving to the inner membrane of the mitochondria to witness the most spectacular piece of molecular engineering in the known universe. Here, we will see how a flow of electrons creates a literal dam of energy, which in turn powers a shimmering, rotating turbine that builds the currency of life at a rate of thousands of molecules per second. To understand this process, I want you to first visualize the structure of the mitochondria. In our previous lesson, we were in the matrix, the very center. Now, we are looking at the inner mitochondrial membrane, which is folded into many beautiful ridges called cristae. Embedded within this membrane is a series of 4 massive protein complexes, numbered simply 1 through 4. Together, they make up the electron transport chain. The process begins when NADH arrives at complex 1. It hands over its high-energy electrons, becoming NAD+ once again, ready to return to the citric acid cycle for more. As these electrons enter the chain, they move from one complex to the next, much like water flowing down a series of cataracts in a river. They move to a mobile carrier called ubiquinone, then to complex 3, then to another carrier called cytochrome C, and finally to complex 4. But these electrons aren't just moving for the sake of movement. As they pass through these complexes, they provide the energy needed to do a very specific type of work. They power pumps that grab protons, which are simply hydrogen ions, or H+, from the matrix, and shove them across the membrane into the narrow space between the inner and outer membranes. Take a moment to let that idea find its place. One with every electron that passes through the chain, the concentration of protons in that tiny intermembrane space grows higher and higher. You are building a proton motive force. Eventually, the electrons reach the end of the line at complex 4. They have given up most of their energy to pump those protons, and they need somewhere to go. This is where oxygen, O2, makes its essential appearance. Oxygen is the final electron acceptor. It sits at the end of the chain, catches the weary electrons, and combines with some nearby protons to form H2O, simple, harmless water. This is the ultimate destination of the air you breathe. Now, let us look at the dam we have created. The intermembrane space is now packed with protons that are desperate to get back into the matrix. They are driven by two forces. They want to move from a high concentration to a low one. And because they are positively charged, they are attracted to the more negative environment inside. However, the membrane is a solid wall that they cannot cross on their own. There is only one gate through which they can return, and it is a magnificent enzyme called ATP synthase. As the protons flow back through this gate, they don't just pass through. They actually cause a portion of the enzyme to rotate, much like water turning a turbine in a hydroelectric dam. This physical, mechanical rotation provides the energy needed to take an ADP molecule and a stray phosphate and snap them together to create ATP. This process, where the movement of ions drives the synthesis of ATP, is called chemiosmosis. If your mind is still following along, I'd try visualizing this shimmering rotating motor. It is the most efficient engine ever discovered, turning nearly 100% of the energy from the proton flow into chemical power. For every check of NADH we cache, we produce roughly 2.5 molecules of ATP. For every FADH2, we produce about 1.5. In total, this grand process yields about 26 to 28 molecules of ATP for every single molecule of glucose, a massive increase over the measly 2 we earned in glycolysis. To truly appreciate the wonder of oxidative phosphorylation, we should look back at how long it took us to understand it. For decades, biochemists were looking for a high-energy intermediate, a specific chemical that linked the electron chain to the production of ATP. They couldn't find one. And it wasn't until 1961 that a British scientist named Peter Mitchell proposed the chemiosmotic hypothesis. Mitchell suggested that the link wasn't a chemical at all, but a physical gradient of protons. At the time, his idea was seen as radical and was met with great skepticism. But as the evidence mounted, the scientific world realized he was right. He showed us that the cell isn't just a test tube of reactions. It is a complex, three-dimensional landscape where space and boundaries are just as important as the chemicals themselves. For this profound insight, he was awarded the Nobel Prize in 1978. If your mind is wandering, try to picture the scale of this. In a single one of your heart cells, there may be thousands of mitochondria, each with thousands of these ATP synthase motors, and they are spinning at speeds of up to 100 revolutions per second. This is not a slow, clunky process. It is a high-speed, precision-tuned hum of activity that occurs as long as you are alive. There is also a fascinating layer of detail regarding uncoupling. In certain tissues, like the brown fat found in newborn babies and hibernating animals, there is a protein called thermogenin. This protein creates a leak in the mitochondrial dam. It allows protons to flow back into the matrix without going through the ATP synthase turbine. Because the energy of the proton gradient isn't being used to make ATP, It is instead released as pure heat. This is a deliberate biological choice to favor warmth over power, ensuring that a baby stays warm even if it cannot shiver. You don't worry about the complex names of the electron carriers like iron-sulfur clusters or flavin mononucleotide. Instead, just focus on the dam and the turbine. The electron transport chain builds the dam, and chemiosmosis uses the turbine. It is a beautiful two-act play that represents the pinnacle of energy efficiency in the natural world. The relevance of oxidative phosphorylation is a matter of life and breath. When we speak of metabolic poisons, we are often talking about substances that interfere with this specific chain. Cyanide, for instance, is so deadly because it binds irreversibly to complex 4. It effectively clogs the end of the electron transport chain. When the electrons can no longer reach oxygen, the whole chain grinds to a halt. The protons stop pumping, the dam empties, the turbines stop spinning, and then the cell can no longer produce the ATP it needs to survive. It is a stark reminder that your life is a constant, high-speed flow. If the flow stops for even a few minutes, the flame goes out. So you see, this isn't just a theory in a book. It explains why we need oxygen so desperately. Oxygen isn't just good for you. It is the essential garbage collector for your spent electrons. Without it, your entire energy economy collapses. This synthesis of breathing and power is why deep rhythmic breathing can be so meditative and restorative. You are literally providing the final ingredient for your cellular turbines to run at their peak efficiency. Everything we have discussed, the glucose, the glycolysis, the Krebs cycle, has been leading to this. It is the ultimate purpose of your metabolism. Even as you lie here now, perfectly still, your mitochondria are processing the remains of your day's meals. They are cashing the checks of your dinner to build the ATP that will repair your muscles, consolidate your memories, and keep your heart beating through the night. You are a shimmering network of tiny hydroelectric dams, all working in silent harmony. We have now reached the summit of our study of energy production. We have watched the electrons flow, seen the protons build their gradient, and marveled at the rotation of the ATP synthase turbine. We have seen the big payout that sustains the complexity of your life. But as we sit here at the end of the 12th lesson, we must recognize that a healthy economy needs more than just a way to spend money. It needs a way to manage its reserves. We have talked about how we burn sugar, but what happens when the sugar runs out? What happens when the cell needs to make glucose from scratch to keep the brain fueled during a long rest or a period of activity? Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we turn our attention to the body's backup plan. We are going to explore the world of gluconeogenesis and glycogen metabolism. We will see how the liver acts as a wise guardian, storing sugar when it is plentiful and building it from scratch when it is scarce, ensuring that the lights never go out in the city of your mind. Having now grasped how we harvest energy, we are perfectly prepared to see how we manage our wealth. So when we return, we will begin our study of gluconeogenesis and glycogen metabolism. Take a long or slow breath. A breath that is currently providing the oxygen for your own shimmering turbines. Your dam is full. Your energy is being replenished, and your body is at rest. I'll be here to show you how you manage your metabolic stores when you are ready. Lesson 13, Gluconeogenesis and Glycogen Metabolism. In our previous lessons, we have spent much of our time admiring the incredible efficiency of the downhill journey. We have watched as your body takes the energy from a meal and dismantles it through glycolysis and the citric acid cycle to fill the shimmering dams of the mitochondria. We have seen how you harvest the fire of the sun to power your every movement. But as we move into this 13th lesson, we must confront a simple reality of life. We do not always have a fresh meal at hand. There are times of rest, times of fasting, and long hours of sleep where the external supply of glucose stops flowing. Yet, your brain never sleeps, and your red blood cells never stop their rhythmic circulation. These parts of you are glucose dependent. They demand a steady, unwavering supply of sugar to function. Tonight we explore the body's backup plan. We will study how the liver acts as a wise and generous guardian, managing the short-term savings account of glycogen, and when necessary, performing the miraculous feat of building new sugar from scratch. In a university curriculum, this is titled Gluconeogenesis and Glycogen Metabolism, and it is the story of how you maintain your internal constancy even when the world outside is quiet and still. To understand how you survive the hours between dinner and breakfast, we must first look at your primary storage system. When you have an abundance of glucose after a meal, your body doesn't simply let the excess drift away. Instead, it carefully packs it into a storage molecule called glycogen. I want you to imagine a large bushy shrub. Each leaf on that shrub is a single molecule of glucose. These leaves are linked together in long chains by alpha-1,4 glycosidic bonds. But every 10 leaves or so, a new branch sprouts out, held by an alpha-1,6 glycosidic bond. This highly branched structure is a masterpiece of biology. Because it has so many ends or tips, your enzymes can snip off hundreds of glucose molecules at once the moment you need a quick burst of energy. The creation of this shrub is called glycogenesis, and its breakdown is called glycogenolysis. Most of this storage happens in two places, your muscles and your liver. However, there is a beautiful altruistic difference between the two. Your muscles are somewhat selfish with their glycogen. They store it only for their own use during exercise. But your liver is the community provider. It stores glycogen specifically to release it back into the bloodstream, ensuring that your brain and other organs stay fueled while you rest. It is the steady hand that balances your blood sugar levels throughout the night. Take a moment to let that idea find its place. As you lie here now, your liver is quietly pruning its glycogen shrubs, sending tiny messengers of energy into your blood to keep your thoughts and your heartbeat steady. However, your glycogen stores are limited. They can only sustain you for about 12 to 18 hours of fasting. When that savings account begins to run low, the body must turn to its second, more complex strategy, gluconeogenesis. The name itself tells the story. Gluco for glucose, neo for new, and genesis for creation. This is the process of making new glucose from non-carbohydrate sources. It is as if, having run out of wood for the fire, you began to build new logs out of the stones and scraps found around the workshop. The primary stones used for this building process are lactate, which comes from your muscles, amino acids, which come from protein breakdown, and glycerol, which comes from the breakdown of fats. This process happens almost exclusively in the liver. It is a series of chemical reactions that looks at first glance like glycolysis running backward. But as we will see, it is not quite that simple. Life never takes the easy way back. It builds a new path that is energetically favorable. By the end of this process, the liver has successfully synthesized a brand new molecule of glucose and released it into the world in a triumph of chemical creativity over scarcity. To truly appreciate the how of gluconeogenesis, we must look at the thermodynamic barriers that exist within the cell. You might remember from our study of glycolysis that 3 of the 10 steps were irreversible. They were like one-way trapdoors that opened with a large release of energy. Because those doors only open one way, the liver cannot simply run the glycolysis movie in reverse to make sugar. Instead, it has to find bypasses, specialized enzymes that act like secret staircases to get around those one-way doors. The first and most difficult bypass happens at the very end of glycolysis, where we must turn pyruvate back into phosphoenol pyruvate, PEP. This is such a difficult uphill climb that it requires 2 separate enzymes and the investment of a significant amount of energy. First, an enzyme called pyruvate carboxylase, which uses the vitamin biotin as a helper, adds a carbon to pyruvate to make oxaloacetate. Then, another enzyme called PEP carboxygenase turns that oxaloacetate into the PEP we need. If your mind is still following along, try visualizing the cost of this production. To break down 1 molecule of glucose in glycolysis, we earn 2 ATP. But to build 1 molecule of glucose in gluconeogenesis, the liver must spend the equivalent of 6 ATP molecules. It is a luxury process, a high-cost manufacturing effort that the body only undertakes because the survival of the brain is worth any price. It is a testament to the hierarchy of your biology. The liver will gladly bankrupt its own energy stores to ensure the command center stays online. Historically, our understanding of this balance was greatly advanced by the work of Carl and Gertie Corey, a husband and wife team who won the Nobel Prize in 1947. They discovered what we now call the Corey cycle. They showed that when your muscles work hard and produce lactate, that lactate isn't just waste. It travels through the blood to the liver, where the liver uses gluconeogenesis to turn that waste back into glucose. The liver then sends that glucose back to the muscle to be used again. It is a perfect circular partnership between the worker, the muscle, and the provider, the liver. This entire system is governed by a pair of opposing hormonal conductors, insulin and glucagon. I like to think of insulin as the whisper of the feast. When you eat, insulin tells the liver to stop making sugar and start storing it as glycogen. But as you sleep and your blood sugar dips, your pancreas releases glucagon, the shout of the fast. Glucagon travels to the liver and activates the enzymes for both glycogen breakdown and gluconeogenesis. It is a constant shifting balance, a molecular tug-of-war that keeps your internal environment in a state of perfect golden equilibrium. Don't worry about the complex names of every bypass enzyme like fructose 1,6-bisphosphatase. Just remember that the liver has a specialized toolkit that other cells do not. Most of your cells can burn sugar, but only the liver and a tiny bit of the kidney has the master key, an enzyme called glucose 6-phosphatase that allows it to actually release that sugar into the bloodstream for others. Without that one specific enzyme, your liver would be a hoarder of energy. With it, it is a philanthropist. The relevance of these storage and manufacturing pathways is something you experience every single night of your life. During the first few hours of sleep, you are primarily fueled by the breakdown of your liver's glycogen. It is your quick-release energy. But as the night moves towards the early morning hours, your glycogen stores begin to dwindle, and gluconeogenesis slowly ramps up to take its place. By the time you wake up, a significant portion of your blood sugar is new sugar, manufactured by your liver while you were dreaming. So you see, this isn't just a theory in a book. It is the principle that explains why you don't wake up in the middle of the night with a crashing headache or a sense of panic from low blood sugar. Your body has a built-in cruise control. It also explains why a healthy liver is so vital to your overall stamina. If your liver cannot store glycogen properly or perform gluconeogenesis efficiently, your energy levels will become volatile, leaving you feeling exhausted after only a short period without food. This synthesis of saving and making energy shows the profound resilience of the human body. We are not fragile machines that stop working the moment the fuel line is cut. We are adaptive, self-sustaining organisms with multiple layers of redundancy, and even the waste from your muscles and the scraps from your fat cells are seen as valuable resources by the liver, which can stitch them back together into the golden fuel of glucose. It is a beautiful lesson in the value of every part of ourselves. Nothing is truly wasted in the economy of the cell. As you rest tonight, your liver is performing its quiet, essential duty. It is monitoring the bank account of your glycogen, and with steady and patient hands, building the new molecules that will greet you when the sun rises. You are being cared for from within by a system that is planned for every eventuality, ensuring that you remain whole and powered through the dark. We have now explored how the body manages its sugar wealth, how it builds the shrub of glycogen for short-term savings, and how it manufactures new glucose through the high-cost bypasses of gluconeogenesis. We've seen the liver act as the grand provider for the glucose-hungry brain. But while glucose is the preferred fuel for the mind, it is not the only energy source the body has. In fact, for long-term survival and for the heavy-duty work of many other organs, the body relies on a much more concentrated and abundant form of fuel. We have discussed the sugar, but we have yet to look at the oil. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we turn our attention away from the carbohydrates and toward the long, energy-rich chains of the lipids. We are going to explore how we tap into our vast oil reserves and how we break down fats to provide the steady, slow-burning fire that sustains us during long periods of rest or activity. Having now grasped the management of our sugar, we are perfectly prepared to appreciate the power of our fats. So when we return, we will begin our study of fatty acid oxidation and lipid catabolism. Take a deep, slow breath. Your sugar levels are steady, your liver is at work, and your body is in perfect balance. I'll be here to show you how you burn your deeper reserves when you are ready. Lesson 14, Fatty Acid Oxidation and Lipid Catabolism. In our previous session, we watched as the liver performed the heroic task of maintaining our blood sugar levels, ensuring that the brain and red blood cells never face a moment of scarcity. We saw the checking account of our glycogen reserves and the manufacturing plant of gluconeogenesis. But as we move into this 14th step of our curriculum, we must address the vast golden reserves of energy that lie deeper within us. If carbohydrates are our quick access cache, then lipids, specifically our body fat, are our long-term investment portfolio, our deep storage gold. In a university biochemistry curriculum, this lesson is titled Fatty Acid Oxidation and Lipid Catabolism. Tonight we are going to explore how we tap into these dense, energy-rich molecules. We will follow the journey of a fat molecule from its quiet resting place in our adipose tissue to its final destination inside the mitochondrial furnace. We will see how your body activates these long chains of carbon and how it methodically clips them into pieces that can feed the citric acid cycle we studied together. It is a story of incredible efficiency, allowing us to survive for weeks or even months on the energy we carry within. Let us settle in and explore the slow-burning fire of lipid metabolism. To understand lipid catabolism, we must first return to the molecule we met in Lesson 7, the triacylglycerol. You might remember it as a glycerol backbone with 3 long fatty acid tails. When your body decides it is time to use these reserves, perhaps because you are in a deep sleep and your carbohydrate levels are low, it begins a process called lipolysis. Specialized enzymes called lipases act like molecular scissors, snipping the 3 fatty acid tails away from the glycerol backbone. The glycerol travels back to the liver to be turned into sugar, but the fatty acids are destined for a much more energetic fate. These fatty acid tails are long, straight chains of carbon atoms, often 16 or 18 carbons long, completely saturated with hydrogen. Because they have so many hydrogen atoms, they are highly reduced, which in the language of chemistry means they are packed with potential energy just waiting to be released through oxidation. However, a fatty acid cannot simply walk into the mitochondria and begin to burn. It is too stable, too quiet. The first thing the cell must do is activate it. In the cytoplasm, an enzyme attaches a molecule of coenzyme A to the end of the fatty acid, creating fatty acyl-CoA. This process is expensive. It costs the cell 2 ATP equivalents, but it is a necessary investment to prime the fat for the work ahead. Once activated, we encounter a physical problem. The long fatty acyl-CoA molecule is too large to cross the inner membrane of the mitochondria on its own. It reaches the gate but finds it locked. To get across, it must use the carnitine shuttle. Think of carnitine as a specialized ferry or a bus. The fatty acid is handed over to a carnitine molecule, carried across the membrane, and then handed back to a fresh coenzyme A on the inside. In this shuttle is the master regulator of fat burning. If the shuttle is busy or blocked, the fat stays in the cytoplasm, unable to be used for fuel. Take a moment to let that idea find its place. Your body is constantly ferrying these long carbon chains across a microscopic border, ensuring that the fuel reaches the furnace only when it is needed. Once inside the mitochondrial matrix, the real work begins. This process is called beta oxidation. It is a beautiful repeating cycle of 4 steps: oxidation, hydration, oxidation, and finally, thiolysis. I want you to imagine a long piece of ribbon. Every time the cycle turns once, a pair of scissors snips exactly 2 carbons off the end of that ribbon. These 2 carbons are released as acetyl-CoA, which you will remember as the golden ticket that enters the citric acid cycle. If your mind is still following along, try visualizing the repetition. If we have a 16-carbon fatty acid, the cycle turns 7 times. Each turn produces 1 molecule of FADH2, 1 molecule of NADH, and eventually 8 molecules of acetyl-CoA. It is a methodical, rhythmic chipping away at the long chain until nothing is left but energy and water. To truly appreciate the logic of beta oxidation, we should look back at how we discovered it. In the early 1900s, a scientist named Franz Knupp performed a clever and elegant experiment. He fed dogs fatty acids that had been tagged with a benzene ring at one end. He found that if he fed them a chain with an even number of carbons, they excreted one type of byproduct. And if the chain was odd, they excreted another. From this simple observation, he deduced that the body must be breaking down fats 2 carbons at a time. This was a revolutionary insight, discovered long before we had the powerful microscopes or chemical sensors we use today. If your mind is still following along, let us consider the sheer density of this energy. You may have noticed that a gram of fat contains 9 calories, while a gram of carbohydrate contains only 4. In biochemistry, we understand exactly why this is. Carbohydrates are already partially oxidized. They have oxygen atoms built into their rings. But fatty acids are almost pure carbon and hydrogen. Because they are so reduced, they have much more room to be oxidized. Furthermore, because fats are hydrophobic, they are stored dry. Carbohydrates, being hydrophilic, must be stored with a great deal of water. If you tried to store all the energy of your body fat as glycogen instead, you would weigh hundreds of pounds more than you do now. Fat is the ultimate travel-friendly fuel, lightweight, compact, and incredibly powerful. There is also a fascinating overflow system we should discuss. Sometimes, if you have been fasting for a long time or are on a very low carbohydrate diet, the liver produces acetyl-CoA through beta oxidation faster than the citric acid cycle can process it. When this happens, the excess acetyl-CoA is converted into ketone bodies. These molecules, like acetoacetate and beta-hydroxybutyrate, are water-soluble and can travel through the blood to reach the brain. Don't worry about the complexity of the names. Just think of ketones as emergency glucose substitutes. While the brain prefers sugar, it is perfectly capable of running on ketones when necessary. This is an incredible evolutionary adaptation, allowing our ancestors to remain sharp and alert even during long periods of famine. It is also important to note how this process is regulated. The cell uses a molecule called malonyl-CoA to keep things in balance. When your body is busy building new fats, after a large meal, malonyl-CoA levels rise and physically block the carnitine shuttle. This ensures that you aren't trying to burn fat at the very same time you're trying to build it. It is a simple, elegant on-off switch that prevents metabolic confusion. The relevance of lipid metabolism is visible in every long-term survival strategy in nature. Consider a bear hibernating through a long frozen winter. For months, the bear does not eat or drink. It survives entirely on the beta oxidation of its stored body fat. This process provides not only the ATP needed to keep the heart beating, but also the metabolic water created at the end of the electron transport chain. The bear literally drinks the byproduct of its fat burning. So you see, this isn't just a theory in a book. It is the principle that explains why you can sleep through the night without feeling the pangs of hunger. During your rest, your body gently shifts its preference. While you are awake and active, you might rely more on the quick burn of glucose. But as you settle into a deep sleep, your growth hormones rise and signal your fat cells to release their stores. Now you become a fat-burning machine while you dream, providing a steady low-intensity flame that repairs your tissues and maintains your temperature. This synthesis of efficiency and endurance shows that our fat is not something to be viewed with disdain. It is a remarkable biological achievement. It is our buffer against the volatility of the world. It is what allows a marathon runner to keep moving at the 20-mile mark long after their glycogen is gone. It is what allows your heart, which prefers fatty acids over glucose as its primary fuel, to beat billions of times without ever taking a break. Your heart is perhaps the most diligent fat burner in your entire body, using the steady energy of beta oxidation to ensure it never tires. As you lie here now, this slow-burning fire is sustaining you. Your fatty acids are being ferried into your mitochondria, clipped into 2 carbon pieces, and turned into the power that keeps your lungs expanding and your heart rhythmically pumping. You are being fueled by the deep, ancient reserves of your own biology. We have now explored the long-term energy stores of the body. We've seen how we activate fatty acids, shuttle them into the mitochondria, and clip them into acetyl-CoA through the repeating cycle of beta oxidation. We have seen how fat provides the endurance and the water we need to survive the long quiet of the night. But our study of the big 3 fuels is not yet complete. We have talked about the sugars, and we have talked about the oils. Now, we must address the most complex and versatile building blocks of all, the proteins. While we usually try to save our proteins for building muscles and enzymes, there are times when the body must tap into them for energy, or simply manage the waste created when old proteins are recycled. Let's hold that thought here. A complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we turn our attention to the unique challenges of nitrogen. We are going to explore how we break down amino acids, and more importantly, how we safely dispose of the toxic ammonia that is created in the process. Having now grasped the oil of our metabolism, we are perfectly prepared to understand the bricks and their unique chemistry. So when we return, we will begin our study of nitrogen metabolism and the urea cycle. Take a deep, slow breath and feel the steady warmth of your internal fire. Your energy is secure, your reserves are being managed with wisdom, and your body is at peace. I'll be here to show you how you handle the chemistry of nitrogen when you are ready. Lesson 15. Nitrogen metabolism and the urea cycle. In our previous explorations, we have spent much of our time admiring the carbon and hydrogen skeletons of the world, the sugars that spark our energy, and the long, oily chains of fats that sustain us through the long night. We have watched the body burn these fuels with incredible efficiency. But as we reach this 15th lesson, we must address the third pillar of our biology, the proteins. Unlike carbohydrates and lipids, which are composed almost entirely of carbon, hydrogen, and oxygen, proteins carry a very special and sometimes stubborn guest, nitrogen. Nitrogen is the essential element that allows us to build the amino acids we studied in Lesson 3. But it presents a unique chemical challenge. When we break down proteins for energy, or simply recycle old ones, we are left with nitrogen-based waste that the body cannot simply burn away into carbon dioxide. Tonight, we are going to explore the sophisticated plumbing of the cell. We will look at how the body strips nitrogen away from amino acids, and most importantly, how the liver performs the vital task of converting toxic ammonia into a safe water-soluble molecule called urea. It is a story of biological detoxification and the careful management of the elements that make us who we are. To understand nitrogen metabolism, we must first recognize that nitrogen is both a treasure and a burden. We need it to build our muscles, our enzymes, and our DNA. But free nitrogen in the form of ammonia is incredibly dangerous to our cells, particularly our neurons. When the body has an excess of amino acids, perhaps from a high-protein meal, or because it is breaking down muscle tissue during a fast, it must remove the amino group NH2 to use the remaining carbon skeleton for energy. The first step in this process is a beautiful, cooperative handoff called transamination. Imagine a crowded room where many people are holding on to a heavy, somewhat awkward package, the amino group. Instead of everyone trying to deal with their own package, they all pass them to a single, designated collector. In most of your cells, that collector is a molecule called alpha-ketoglutarate. When an amino acid hands its nitrogen to alpha-ketoglutarate, the amino acid becomes a keto acid, which can be burned for fuel, and the collector becomes glutamate. This glutamate then travels through the blood to the liver, which is the grand processing center for nitrogen. Once inside the liver, the process of oxidative deamination occurs. The nitrogen is finally released from the glutamate, but it is released as ammonia, NH3. Ammonia is highly alkaline and toxic. If it were allowed to build up, it would disrupt the delicate pH balance of your brain and cause profound confusion. Take a moment to let that idea find its place. Your body has a constant, invisible toxic spill occurring every time you process protein, and it has developed a masterful way to contain and neutralize that spill before it can ever do harm. To solve this ammonia problem, the liver uses the urea cycle. This is a 5-step metabolic pathway that takes that dangerous ammonia and packages it into urea. Urea is a neutral, harmless molecule that your kidneys can easily filter into your urine. The cycle begins in the mitochondria of the liver cells, where the ammonia is combined with carbon dioxide and a phosphate to create a molecule called carbomoyl phosphate. This is the priming step, much like we saw in glycolysis. Once primed, the carbomoyl phosphate enters the cycle by joining with a molecule called ornithine to create citrulline. Citrulline then leaves the mitochondria and enters the cytoplasm, where it undergoes a series of transformations. It joins with an amino acid called aspartate to form argineosuccinate, which is then split to release fumarate and leave behind arginine. Finally, an enzyme called arginase snips the urea molecule off the arginine, which releases the urea into the blood and regenerates the original ornithine, ready to start the cycle all over again. If your mind is still following along, try visualizing this as a continuous 5-room factory line. The toxic ammonia enters through the back door, is strapped to a carrier, moved through several workstations, and finally comes out the front door as a safe, boxed-up product called urea. The carrier, the ornithine, never leaves the factory. It simply walks back to the starting line to pick up the next package. To truly appreciate the elegance of the urea cycle, we should look at its history and its deep connection to the other cycles we have studied. In 1932, 5 years before he discovered the citric acid cycle, Sir Hans Krebs, along with his student Kurt Henseleit, mapped out this pathway. It was actually the very first metabolic cycle ever discovered. It showed the scientific world that metabolism wasn't just a series of straight lines, but a system of rotating wheels. But there is an even more profound layer to this story. In university lectures, we often speak of the Krebs bicycle. If you look closely at the urea cycle, you'll notice a molecule called fumarate is released in the 4th step. If that name sounds familiar, it is because fumarate is also an essential intermediate in the citric acid cycle, lesson 11. This means the two cycles are physically and chemically linked together, like two wheels of a bicycle. The waste from the urea cycle, the fumarate, is actually fuel for the energy-producing citric acid cycle. This connection is a masterpiece of efficiency. The energy released by the citric acid cycle helps provide the ATP needed to power the urea cycle, and the byproducts of nitrogen disposal help keep the energy engine turning. Nothing is wasted. Everything is interconnected. It is as if the exhaust from your car's engine was captured and turned back into a small amount of gasoline. If your mind is wandering, try to picture the nitrogen paradox. Nitrogen is so stubborn that it takes massive amounts of energy for bacteria in the soil to fix it into a form we can eat. Yet once it is inside us, it is so reactive that we must spend even more energy, 4 high-energy phosphate bonds per turn of the cycle, just to safely get rid of the excess. We are constantly paying an energy tax to manage the nitrogen that builds our physical structure. Don't worry about memorizing the names like citrulline or argininosuccinate right now. Instead, just focus on the wisdom of the liver. The liver is the only organ in your body that has all 5 enzymes of the urea cycle. It is the only place equipped to handle this specific type of hazardous waste. This is why your liver is so central to your overall health. It is the guardian that prevents the byproducts of your own muscles and your own food from poisoning your mind. The relevance of nitrogen metabolism is something you see in both medicine and everyday health. Consider someone who goes on an extremely high-protein diet. Their liver must work overtime to process the increased influx of nitrogen, producing much higher levels of urea. This is why it is so essential to drink plenty of water when increasing protein intake. The kidneys need that water to flush the increased urea out of the system. So you see, this isn't just a theory in a book, and it explains why people with liver challenges often experience brain fog or confusion. If the liver cannot perform the urea cycle effectively, ammonia levels in the blood begin to rise. This ammonia crosses the blood-brain barrier and interferes with the way brain cells communicate. In a very real sense, the clarity of your thoughts right now is dependent on the quiet, rhythmic turning of your liver's urea cycle. You can also find the synthesis of this lesson in the world of skincare. You may have noticed that many high-quality moisturizers contain urea as an ingredient. It might seem strange to put a waste product on your face, but in biochemistry, urea is a powerful humectant. It is a molecule that loves water so much that it pulls moisture from the air and traps it in the skin. Your body actually uses small amounts of urea in your skin and your kidneys to manage water balance. What is waste in one context is a precious tool in another. Everything we have discussed, the transamination handoffs, the toxic ammonia, the 5-step cycle, is a reminder of the incredible balance of your life. You are a system that is constantly cleaning itself, managing its resources with a level of care that we are only just beginning to understand. As you rest tonight, your liver is performing these thousands of handoffs, catching the nitrogen from your day's activities and boxing it up for removal, ensuring that when you wake up, your internal environment is fresh, clean, and clear. We have now explored the sophisticated plumbing and detoxification systems of the cell. We've seen how we manage the stubborn guest of nitrogen, how we pass the baton of amino groups to the liver, and how the Krebs bicycle links our waste disposal to our energy production. We have seen the cleanup crew of the cell. But as we sit here at the end of the 15th lesson, we have finished our study of the fuels and the building materials of the body. We have talked about the sugars, the fats, and the proteins. We have seen how we burn them, build them, and clean up after them. But there is one final, profound question. Where did the instructions for all of this come from? How does the liver know how to build the enzymes for the urea cycle? How do the mitochondria know how to build the electron transport chain? Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we return to the very heart of the cell. We're going to go back to the master blueprints we introduced in Lesson 8. But this time, we aren't just looking at their structure. We are going to watch them in action. Having now grasped how the body works, we are perfectly prepared to see how it remembers how to work. Our next step takes us into the world of DNA replication and repair. We will see how life copies its vast library of knowledge without making a single mistake, ensuring that the dance of biochemistry can continue for another day and another generation. Take a long, slow, deep breath. Your internal world is clean, your nitrogen is balanced, and your body is at rest. I'll be here to show you the master architects of your code when you are ready. Lesson 16. DNA Replication and Repair. We have spent much of our journey exploring the stuff of life, the fuels we burn, the boundaries we build, and the intricate plumbing that keeps our internal environment clean. We have seen how the body performs 1,000 different tasks with unfailing rhythm. But as we step into this 16th lesson, we must return to the most sacred space in the cell, the library of the nucleus. You see, a city that functions perfectly today must also have a way to ensure it functions tomorrow. It must be able to pass its wisdom, its blueprints, and its very identity to the next generation of cells. In our 8th lesson, we marveled at the twisting double helix architecture of DNA. Tonight we are going to watch that architecture in motion. In a university biochemistry curriculum, this is the study of DNA replication and repair. We are going to explore how life makes a perfect copy of its vast library, billions of characters long, without losing a single word of the story. We will see the master builders at work and the diligent editors who check every line for errors. It is a story of incredible fidelity and the profound biological commitment to accuracy. To understand DNA replication, we must first appreciate the elegant logic of its structure. Because adenine always pairs with thymine, and cytosine always pairs with guanine, each strand of the double helix contains all the information necessary to rebuild its partner. When the cell prepares to divide, it doesn't need to write a new book from scratch. It simply needs to unzip the old one and use each half as a template. This is what we call semi-conservative replication. Take a moment to let that idea find its place. Every single strand of DNA in your body is half old and half new. You carry within you physical pieces of the original code that built you as a child, intertwined with the new strands created just hours ago. The process begins at a specific location called the origin of replication. An enzyme called helicase arrives and begins to unzip the double helix, breaking the gentle hydrogen bonds between the bases. This creates a Y-shaped structure known as the replication fork. Because the DNA is a tightly twisted spiral, unzipping it creates a lot of tension further down the line, much like pulling apart the strands of a rope. To prevent the DNA from knotting up, an enzyme called topoisomerase works ahead of the fork, snipping and rejoining the strands to relieve the pressure. Once the strands are separated, we meet the master builder, DNA polymerase. However, DNA polymerase is a very particular worker. First, it cannot start a new strand from nothing. It needs a small starter piece to hold onto. An enzyme called primase lays down a short stretch of RNA called a primer to give the builder a place to begin. Second, DNA polymerase can only move in one direction. It only builds the new strand from the 5' end to the 3' end. This creates a fascinating challenge at the replication fork. One strand, which we call the leading strand, runs in the perfect direction for the builder. DNA polymerase can simply follow the helicase, building one long, continuous piece. But the other strand, the lagging strand, runs in the opposite direction. The builder must work backward away from the fork. If your mind is still following along, try visualizing this. It is like a person trying to paint a long fence while walking backward. Every few feet, they have to stop, jump back toward the gate, and paint another section toward where they just were. These short, fragmented pieces of DNA on the lagging strand are called Okazaki fragments. Eventually, another enzyme called DNA ligase comes along and glues all these fragments together, creating a solid, continuous strand. By the end of this process, the cell has 2 identical double helices, where there was once only one. To truly appreciate the wonder of replication, we must look at its accuracy. The human genome contains roughly 3 billion base pairs. If you were to type 3 billion characters on a keyboard, and even the most skilled typist would make thousands of mistakes. Yet, DNA polymerase is so precise that it makes an error only about once every 10 to the 7th power bases. But even that isn't good enough for life. Through a second layer of protection called proofreading, the error rate is dropped to a staggering 1 in a billion, 10 to the power of minus $9. DNA polymerase has a built-in delete key. If it accidentally adds the wrong base, say, placing a cytosine where a thymine should be, the enzyme feels the bump in the helix. It immediately stops, moves backward, using its exonuclease activity to snip out the wrong base, and then tries again. It is a level of self-correction that ensures your genetic identity remains stable across decades of life. Historically, our understanding of this semi-conservative nature was proven by a most beautiful experiment performed by Matthew Meselson and Franklin Stahl in 1958. They used different isotopes of nitrogen to weigh the DNA of bacteria as they reproduced. By watching how the heavy and light DNA mixed over generations, they proved that the strands were indeed being split and shared. It showed us that life is a continuous, unbroken chain of information. However, even the best-kept library faces threats from the outside world. Your DNA is constantly being bombarded by ultraviolet light from the sun, chemicals in the environment, and even the reactive oxygen species produced by your own metabolism. This is where DNA repair becomes vital. One of the most common types of damage is caused by UV light, which can make two adjacent thymine bases stick together, forming a kink called a thymine dimer. If your mind is still following along, try visualizing a railroad track where two of the rails have melted together and buckled upward. A train, the DNA polymerase, cannot pass over that buckle. To fix this, the cell uses nucleotide excision repair. A team of enzymes scouts the DNA for these kinks. When they find one, they cut out the damaged stretch. A different polymerase fills in the gap with the correct bases, and ligase seals the deal. Don't worry about memorizing the details of every repair enzyme like MUT-S or MUT-L. Just let the logic of it wash over you. Your cell is not a passive victim of the environment. It is an active, vigilant guardian of its own code. It is constantly scanning its library, looking for torn pages or smeared ink, and fixing them before the information can be lost. This is a good place to rest your thoughts for a moment before we continue. Think about the peace of knowing that even as you sleep, 1,000 microscopic editors are at work, ensuring your story remains true. The relevance of DNA replication and repair is written in the very process of how we grow and how we age. Consider the ends of your chromosomes, which are called telomeres. Because of the way the lagging strand is built, the very tip of the DNA cannot be copied perfectly. A tiny bit is lost every time a cell divides. I like to think of telomeres as the plastic aglets at the end of a shoelace. They prevent the DNA from fraying, but they get shorter with every division. This shortening is one of the primary reasons our cells eventually stop dividing and we begin to age. So you see, this isn't just a theory in a book. The fidelity of your DNA replication is what prevents cancer. Most cancers begin with a failure in these very systems, a mistake during replication that wasn't caught, or a piece of damage that wasn't repaired. By maintaining these repair pathways through healthy habits and proper nutrition, we are quite literally supporting the editors of our life's work. This synthesis of information and stability is what allows life to endure. It is why you look like your parents, and why your children will look like you. It is the reason why a single fertilized egg can become a complex human being with trillions of cells, all carrying the same set of instructions. We are the products of an ancient, high-fidelity copying machine that has been running for billions of years. As you rest tonight, this process is likely happening somewhere in your body. Perhaps in your skin or your bone marrow, cells are quietly unzipping their DNA, building new strands, and checking for errors. You are a work of constant, careful self-replication. You are being renewed letter by letter while you dream. We have now explored the Grand Library's copying room. We've seen the helicase unzip the helix, watched the DNA polymerase build new strands in the 5' to 3' direction, and admired the diligent work of the repair enzymes as they fix the kinks in the code. We have seen how life preserves its past to secure its future. But a library of blueprints, no matter how perfectly copied, is useless if the books never leave the shelf. A blueprint for a house doesn't build the house. A person must read the blueprint and turn those instructions into action. Having now grasped how we copy the code, we are perfectly prepared to see how we read it. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, where we see the cell take a specific gene from the DNA and create a portable temporary copy that can be sent to the workshop. We are going to explore the world of transcription and RNA processing. We will see how life translates the silent wisdom of the DNA into the active messages of RNA. So when we return, we will begin our study of how the code is spoken aloud. Take a deep, slow breath. Your blueprints are safe. Your editors are on duty. And your body is at peace. I'll be here to show you how you read your own story when you are ready. Lesson 17, Transcription and RNA Processing. In our last session, we stood in the quiet sanctuary of the nucleus and watched the incredible feat of DNA replication. We saw how life, with almost impossible precision, makes a faithful copy of its entire library to ensure its future. But a library, no matter how perfectly preserved, serves no purpose if the books never leave the shelves. The DNA is the master architect's original parchment. It is too precious and too fragile to be hauled out to the noisy, bustling construction sites of the cell. To turn a thought into an action, or a blueprint into a building, the cell must create a message, a portable, temporary, and highly specific copy of a single gene. In our university journey, this is the study of transcription and RNA processing. We are going to watch how the cell reads its own wisdom and translates the silent code of the double helix into the active language of RNA. We will see how a raw message is edited, refined, and protected before it is sent out into the world. It is the story of how the word of the genome becomes the flesh of our physical being. To understand transcription, we must first imagine the nucleus as a grand, silent archive where the master blueprints are kept in a vault. When the cell needs a specific protein, perhaps an enzyme to digest your dinner, or a hormone to help you grow, it doesn't copy the whole library. Instead, it finds the specific page or gene it needs and creates a transcript. This transcript is messenger RNA, or mRNA. Unlike the double-stranded DNA we studied, mRNA is a single nimble strand that is designed to be a temporary messenger. The master of this process is an enzyme called RNA polymerase. If DNA polymerase was the copyist for the whole library, RNA polymerase is the scribe for a single story. The process begins at a specific sequence on the DNA called the promoter. Think of the promoter as a large illuminated start sign at the beginning of a gene. RNA polymerase recognizes this sign and sits down upon the DNA. It doesn't need a primer like the replication enzymes did. It is a self-starter. It gently unwinds a small bubble of the DNA, exposing the bases so it can begin its work. Take a moment to let that idea find its place. As you lie here, your DNA is being unzipped in thousands of different locations, not to be copied entirely, but to be read for the specific needs of the moment. Once the enzyme is settled, it moves along the DNA strand, reading the code in the 3' to 5' direction, and building the new mRNA strand in the 5' to 3' direction. As it moves, it follows the same base pairing rules we learned in Lesson 8, with one beautiful exception. Where the DNA code calls for an adenine, the RNA polymerase does not reach for a thymine. Instead, it uses uracil, U. This single letter change acts as a chemical signal to the cell, a way of saying, this is a message, not a permanent record. This stage is called elongation. Imagine the enzyme gliding smoothly down the DNA track, like a train, and trailing behind it, a growing, shimmering ribbon of RNA. As the enzyme passes, the DNA behind it zips back up into its double helix shape, undisturbed and protected. The information has been spoken into the RNA, and the original remains as pristine as it was before. Eventually, the scribe reaches a stop sign in the code, which we call the terminator. At this point, the RNA polymerase lets go of the DNA, and the newly formed strand of RNA, the primary transcript, is released. It is a complete letter-for-letter copy of the gene's instructions. If your mind is still following along, try visualizing this. The cell has taken a high-resolution photograph of one small section of the blueprint. It is a perfect record of the text, but it isn't quite ready for the construction site yet. It is raw, it is vulnerable, and it needs a bit of editing. In the history of biochemistry, we once believed that genes were continuous, like a sentence in a book that begins at the first word and ends at the last. But in 1977, two scientists named Richard Roberts and Philip Sharp made a discovery that shocked the scientific world. They found that in complex organisms like us, the eukaryotes, our genes are interrupted. They are full of sequences that do not actually code for the protein. We call these intervening sequences introns, and the parts that carry the actual instructions are called exons. Imagine you are reading a recipe for bread, but every few sentences, someone has inserted a paragraph about the history of wheat or a description of a sunset. The exons are the instructions for the flour, water, and yeast. The introns are the extra fluff. Before the mRNA can be used, the cell must perform a process called splicing. A massive molecular machine called the spliceosome arrives, snips out the introns, and glues the exons together into one continuous, meaningful sentence. This discovery was so profound that it earned Roberts and Sharp the Nobel Prize. It revealed that the cell is not just a passive reader of the code, it is an active editor. This leads us to one of the most remarkable features of our biology, alternative splicing. By choosing which exons to keep and which to discard, a single gene can produce several different versions of a protein. It is like a director taking the same footage and creating a theatrical cut, a television edit, and a director's cut. This is why we can be so complex even though we have fewer genes than a stalk of corn. We are masters of the edit. But the editing doesn't stop with splicing. Because the cytoplasm is a dangerous place full of enzymes that like to eat RNA, the transcript needs protection. First, the cell adds a 5' cap to the beginning of the strand. Think of this as a hard hat that protects the front of the message and helps the ribosome recognize it later. Then, at the other end, it adds a long tail of adenine bases, known as the poly-A tail. This tail is like a protective buffer or a fuse on a firecracker. As the mRNA travels, the tail is slowly eaten away, but the message in the middle remains safe. Don't worry about memorizing the complex chemistry of the cap or the exact proteins of the spliceosome right now. Instead, just think about the care the cell takes. It doesn't just throw its messages out into the world. It marks the start, it trims the junk, it glues the meaning together, and it protects both ends. This is a good place to rest your thoughts for a moment before we continue. Think about the incredible attention to detail happening within your own cells. Every message is handled with the precision of a royal scroll. The relevance of transcription and RNA processing is found in the very essence of human health and the diversity of life. When these editing systems fail, the results can be profound. For example, some forms of a blood disorder called thalassemia are caused not by a mistake in the DNA text, but by a mistake in the splicing. The spliceosome misses a cut, and a piece of an intron is left in the final message. Because the recipe is now garbled, the cell cannot build the hemoglobin it needs to carry oxygen. So you see, this isn't just a theory in a book. It is the principle that explains why our bodies are so resilient yet so delicate, and it is also the basis for some of the most advanced medicines of our time. You may have heard of antisense oligonucleotides, which are drugs designed to bind to a specific piece of RNA and hide a splice site. This allows us to actually change the way a cell edits its own messages, helping to treat genetic conditions that were once thought untreatable. This synthesis of information and editing shows us that life is a conversation. The DNA provides the vocabulary, but the transcription and processing provide the grammar and the tone. It is through this process that a single cell knows whether to be a muscle cell, a nerve cell, or a skin cell. They all have the same library, but they are all printing different sets of edited messages. As you rest tonight, this conversation is continuing within you. Your cells are reading the blueprints of your life, deciding which stories need to be told to repair your tissues and balance your hormones. You are a living library that is constantly publishing a fresh edition of its own existence, one transcript at a time. We have now explored the scribing and editing rooms of the cell. We've seen how RNA polymerase reads the DNA vault, how the introns are removed to find the meaning, and how the 5' cap and poly A tail provide the protection needed for the journey ahead. We have watched the word become a message, but a message, no matter how perfectly edited or protected, is still just a set of instructions on a piece of paper. It is still just code. For our final lesson, we are going to follow the mRNA as it leaves the safety of the nucleus and enters the bustling workshop of the cytoplasm. We are going to watch as that code is finally turned into a physical, three-dimensional protein. Let's hold that thought here. A complete piece of the puzzle. Our next lesson will hand us the final, adjacent piece, where we turn our attention to the grand finale of the central dogma. We are going to explore the world of translation and the genetic code. We will see the ribosome in action, the towers of tRNA, and the birth of a new protein chain. Having now grasped how we write the message, we are perfectly prepared to see how we build the result. So when we return, we will begin our final study. Take a deep, slow breath. Your messages are sent, your instructions are clear, and your body is at peace. I'll be here to show you the final construction when you are ready. Lesson 18. Protein synthesis and the genetic code. We have reached the final, most profound step of our journey together. In our previous sessions, we explored the silent library of the DNA, and watched as the cell carefully scribed and edited its temporary messages in the form of mRNA. We have seen how life prepares its instructions with meticulous care. But a message is still just a set of symbols, a string of letters on a page. To make a person, to build a heart that beats or a mind that wonders, those symbols must be translated into physical reality. In this 18th lesson, we are going to witness the grand finale of the central dogma, the process of translation. This is the moment where the abstract code of the genetic library becomes the tangible, three-dimensional proteins that perform the work of life. We will see the ribosome, the great molecular factory of the cell, as it reads the mRNA message and assembles amino acids into long, beautiful chains. It is the story of how the invisible word of the genome becomes the physical flesh of your being. To understand translation, we must first recognize that the cell is dealing with two completely different languages. One is the language of nucleic acids, written with the four-letter alphabet of A, U, C, and G. The other is the language of proteins, written with the 20 different amino acids we studied in Lesson 3. To move from one to the other, the cell needs a dictionary and a translator. The dictionary is the genetic code, and the translator is a remarkable molecule called transfer RNA, or tRNA. The genetic code is read in groups of 3 letters, which we call codons. Imagine the mRNA as a long sentence with no spaces between the letters. The cell reads this sentence 3 letters at a time, like a UG-CAG-UUA. Each of these 3-letter words corresponds to a specific amino acid. For example, the codon UUU always tells the cell to add the amino acid phenylalanine. There are 64 possible codons, which is more than enough to cover our 20 amino acids, providing a level of redundancy that protects us from small errors. The actual work of translation happens within a massive molecular machine called the ribosome. I want you to visualize the ribosome as a grand 2-part factory made of protein and ribosomal RNA, rRNA. It has a large subunit and a small subunit that come together like a steady hand gripping the mRNA strand. Inside this factory, there are 3 specific rooms or sites. The A site, the P site, and the E site. Now meet the translator, the tRNA. A tRNA molecule is shaped like a cloverleaf, but it acts like a bilingual bridge. At one end, it has an anticodon, a 3-letter sequence that is the perfect mirror image of an mRNA codon. At the other end, it carries the specific amino acid that matches that codon. It is the only molecule in your body that can speak both languages at once. Take a moment to let that idea find its place. Your body is full of these tiny bridges, each one carrying a single building block, waiting for its turn to speak. The process of building the protein, what we call the translation cycle, happens in 3 rhythmic acts. The first is initiation. The small subunit of the ribosome finds the mRNA and searches for a very specific start signal, the codon AUG. This codon not only tells the factory to start building, but it also calls for the first amino acid, methionine. Once the start signal is found, the large subunit clicks into place, and the factory is open for business. Next comes elongation. This is a beautiful, repeating cycle of addition. A new tRNA, carrying its amino acid, enters the A site of the ribosome. If its anticodon matches the mRNA codon waiting there, it stays. The ribosome then performs a spectacular feat. It snips the growing protein chain away from the previous tRNA and glues it onto the new amino acid using a peptide bond. The ribosome then shifts over by exactly 3 letters. The empty tRNA is kicked out of the E site, and the A site is now open for the next guest. If your mind is still following along, try visualizing the rhythm. Entry, bonding, shifting. Entry, bonding, shifting. This continues codon by codon as the protein chain grows longer and longer, beginning to fold into the shapes we studied in Lesson 4, even as it is being born. Finally, we reach termination. The ribosome encounters a stop codon, UAA, UAG, or UGA. There are no tRNA molecules for these codons. Instead, a release factor enters the ribosome, causing the whole machinery to disassemble and releasing the finished protein into the cell. The message has been delivered. The work is complete. To truly appreciate the wonder of the genetic code, we must look at its history and its profound universality. In the early 1960s, scientists like Marshall Nirenberg and Har Gobind Khorana performed a series of ingenious experiments to crack the code. They created synthetic mRNA strands made only of one letter, like UUUUUU, and watched to see which protein the cell would build. When they saw a chain of pure phenylalanine, they knew they had found the first word of the dictionary. By 1966, they had mapped all 64 codons, revealing the hidden language of all life on Earth. One of the most moving aspects of this code is that it is nearly universal. Whether you are a blue whale, a blade of grass, or a microscopic bacterium, the codon CCG always means proline. This tells us that every living thing on this planet is part of the same ancient conversation. We all use the same dictionary to turn our blueprints into reality. This universality is what allows us to perform modern miracles, like producing human insulin in bacteria. The bacteria read the human instructions because the language is exactly the same. If your mind is still following along, let us consider the wobble hypothesis proposed by Francis Crick. You might have noticed that we have 64 codons, but only about 40 different types of tRNA. This seems like a mismatch. Crick realized that the third letter of the codon-anticodon pairing is flexible, or wobbles. As long as the first 2 letters are a perfect match, the third letter can sometimes be slightly off, and the correct amino acid will still be delivered. This wobble is a clever bit of biological efficiency, allowing the cell to do more with fewer tools while maintaining the accuracy of the message. There is also the question of how the tRNA gets its amino acid in the first place. This happens through a group of enzymes called aminoacyl, tRNA synthetases. I think of these as the ultimate editors. There is one specific enzyme for each of the 20 amino acids. Their job is to grab a tRNA and a matching amino acid and link them together using ATP. They are so precise that if they grab the wrong amino acid, they can actually feel the mistake and correct it before the tRNA is sent to the ribosome. The accuracy of your proteins begins here, with these quiet, diligent matchmakers. Don't worry about memorizing every codon in the table. Just think of the logic. The cell has a start signal to begin the work, a triplet code to ensure diversity, a wobble for efficiency, and stop signals to conclude the task. It is a system of profound mathematical elegance, functioning within you at a scale of billions of transactions per second. Take a deep breath and let the complexity of that harmony settle over you. The relevance of translation is the foundation of modern medicine, particularly in our fight against infection. Many of the most common antibiotics we use today, medicines like tetracycline, erythromycin, and streptomycin, work by targeting the translation machinery. What is truly remarkable is that bacterial ribosomes are slightly different in shape and size than our own human ribosomes. These antibiotics are designed to be molecular monkey wrenches that only fit into the bacterial factory. They jam the ribosome, preventing the bacteria from building the proteins they need to survive, while leaving your own protein synthesis completely undisturbed. So you see, this isn't just a theory in a book. It is the principle that allows us to cure a pneumonia or an ear infection. It is a war of the factories, fought at the level of the codon and the tRNA. We also see the synthesis of this lesson in the field of genetic engineering. Because the code is universal, we can teach cells to do things they have never done before. We can take the gene for a spider's silk and place it into a goat's DNA. And the goat's ribosomes will read that code and produce silk proteins in its milk. This isn't magic. It is simply the natural result of a shared biological language. We are learning to write in the language that nature has been speaking for billions of years. On a more personal level, translation is the process of your own constant renewal. Every time you exercise and your muscles grow stronger, you are witnessing an increase in translation. Every time you learn a new fact and your brain forms a new connection, specific proteins are being translated at your synapses to hold that memory in place. You are a physical being created by a continuous stream of molecular translations. Your thoughts, your strength, and your very presence in this world are the meaning derived from the text of your genome. As you lie here now, in the quiet of the night, this process is reaching its gentle conclusion for the day. Your ribosomes are still clicking along the mRNA strands, building the enzymes that will digest your breakfast and the structural proteins that will repair your skin. You are being built and rebuilt, a one amino acid at a time, with a level of care and precision that is truly breathtaking. We have now completed our grand tour of the molecular world. We have traveled from the liquid cradle of water to the complex engines of metabolism, and finally to the sacred archive of the nucleus. We have seen how the word of the DNA becomes the message of the RNA, and how that message is finally translated into the masterpiece of the protein. This marks the end of our 18-lesson curriculum on biochemistry for sleep. You have walked through the fundamental logic of life itself. You have seen that you are not a random collection of chemicals, but a highly organized, purposeful, and beautiful symphony of molecular interactions, from the spinning turbines of your mitochondria to the wobbling tRNAs in your ribosomes, and every part of you is working in harmony to keep you whole. As you drift off now into a deep and restorative sleep, I want you to carry this sense of wonder with you. You don't need to worry about the details of the codons or the names of the enzymes. Your body has already memorized them. It knows the dance by heart. You can rest in the knowledge that you are perfectly designed and expertly maintained. The same code that builds the stars in the sea is building you right now in the silence. The lesson is complete. The work of the day is done. Sleep deeply, seeker of knowledge. You are a living miracle, and the story of your life is written in the most beautiful language ever known. Good night.